Hi again, everyone. Thank you so much for coming back. I hope everyone had an opportunity to recharge and move around a little bit during the break. So today, one of the very special elements of this event is that we have two very special keynote speakers. We already heard from Susan Manful, and you know, about their beautiful daughter Alex and their very poignant call to action. Our other keynote speaker that I have had the pleasure of knowing now for over a number of years is Nicole Bell. She's the CEO of Galaxy Diagnostics. She wrote a book called What Lurks in the Woods that I had the pleasure of pre-reading. Nicole is going to share her personal story and her journey. That is the reason that led her to want to literally be able to unveil some of these stealth hidden infections in patients that are suffering via her work with the incredible team, Jen Miller, Ed Breitschwert, Ricardo Maggi, Amanda Elam. Forgive me if I'm missing anyone at Galaxy Diagnostics. So with that, I'll hand it over to Nicole.
Thank you, Nikki, and hello, everyone. So, in a day like today, I think images like this are very common, right? Where you're looking at the pathobiology and actual mechanisms leading to decline. I want to talk about something different. I want to talk about the personal story behind these pictures. So I want to introduce you to Russ. So I first met Russ. I did my undergraduate and graduate work at MIT, and my MIT professor recruited me to work at his startup. And when I joined, quickly thereafter, I met Russ, who was one of the executives on the team. And we worked together very closely for about 3 years, and we became best of friends. And eventually we became more than friends. We were kind of an unlikely couple, but we were very well suited together. And we ended up getting married and having two wonderful children, Brian and Haley. And Russ was really a unique individual. He was extremely talented as a musician. He was a world-class guitarist. I show him with an acoustic. He actually preferred electric, a big jazz guy. He also was a brilliant engineer. So he had degrees in electrical engineering and computer science, and that the combination of left brain, right brain really made him extremely creative and good at solving problems. So he had 24 patents on different innovations in technology. He was a leader in his field in electronics. And he was also really an avid outdoorsman. Anything that could get him on a trail, skiing, biking, hiking, whatever it was. And he also had a really quirky and funny sense of humor. So whenever you wanted to find Russ in a crowded room, you literally just had to follow the laughter and you would find him. And we had a fabulous marriage and wonderful family. And then all of a sudden things started to change. He became moody and irritable and angry, and we were fighting all the time, and I couldn't figure out why. And so we went to therapy, and really nothing was getting better. And I talked to him, and the disorders that he was starting to, the depression, the anxiety, it was building. And then finally, I realized that it was more than just the mood-based disorders. He was also forgetting things. He would forget the time to pick up our children from daycare. He would forget the 4-digit security code to our house alarm system. And that was when I realized that there was something more sinister at play. And I tried to convince him to go see a doctor. And so I started researching to prepare for our first doctor's appointment, consulting Dr. Google, right? And one of the first thing that popped up when I started researching brain fog and cognitive decline was Lyme disease. And it immediately resonated with me because it just kind of made sense. Russ was an avid outdoorsman. He had been exposed to multiple ticks, and I personally picked them off of him. And he'd lived in multiple endemic areas. And so it absolutely just seemed to fit. And so we went to the doctor and I said, hey, I would like to test him for this. And I really just want you to help me get my husband back. And so at that first doctor's visit, we went to an integrative physician. And, we really wanted to dig deep because somehow I knew that his case was complicated. And we ran a test for Lyme disease, the standard panel that's recommended by the CDC. And he had the Western blot, and only one of the 5 bands required to call a positive was positive. And so he was considered negative. And she had multiple other pages of testing results. And as she reviewed them, she said, well, they were quote unquote normal, right? And she really didn't have any indication of what was leading to my husband's decline. And so since most of his issues were neurocognitive, she referred us to a neurologist. And when we went to the neurologist, we got a stunning diagnosis, right? They subjected him to the standard array of, cognitive tests, and he failed miserably. I mean, I knew something was wrong, but it was way more wrong than I understood at the time. And that prompted them to get some scans. And when his PET scan results came back, they said that he had a diagnosis of early onset Alzheimer's disease. Excuse me? Okay. And to make matters even worse, when they looked at his scans, they said that his condition and his scans were consistent with late stage disease, right? So here I was still trying to convince him that something was wrong, and doctors were telling me that he was in the late stages of Alzheimer's. We were completely floored. And I'm an engineer, right? Ingrained in me is the ability to make sense of problems, to find root causes. And none of this made sense. So we kept going from doctor to doctor and saying, why is this happening, right? We ran genetic tests. He had 0 genetic markers for early onset Alzheimer's disease. He had no comorbidities that increased the likelihood of Alzheimer's. And he was doing all the things that you're supposed to do for brain health: Mediterranean diet, exercise. He was otherwise very healthy. And we went from doctor to doctor looking for answers and got nothing. And we finally landed in the expert in our area, which was our 4th neurologist. And he gave him all the testing, reviewed the scans, and he looked at us and he said, yep, I agree that it's Alzheimer's disease. Russ stormed out because it was a long day and he was pissed off. And I looked at the doctor and I said, doctor, I know what the diagnosis is. What I don't understand is why. Why is this healthy man with no predisposition to this disease declining so quickly? There has to be a reason. There has to be a root cause. And he looked at me and he said, Mrs. Bell, I know this isn't what you want to hear, but sometimes shit happens. I tell you what, I'm from Boston. It's pretty hard to get me riled up, but I have never wanted to strangle a human being as much as I wanted to strangle that doctor in that moment, right? The fact that that was the best that modern medicine had to offer me was shit happens, while my husband was systematically deteriorating in front of my eyes, was just unacceptable, completely unacceptable. But we had spent over a year going from doctor to doctor trying to get answers, and we weren't finding any. And so at that point, Russ was the one who came to me and said, we have to plan. We have 2 young children. What are we going to do? How are we going to figure this out? And so I essentially gave up and accepted the diagnosis. But then one morning, I got a phone call, and it was from my brother. And it's my older brother. And as it turns out, he had been dealing with a long health issue with his wife. And after years of her struggles, they finally figured out that she had multiple tick-borne infections. And my brother's a doctor, we're very similar, and he started researching, and he's like, Nick, I think this is what's going on with Russ. And I said, I know that was my first instinct, but he tested negative. And then he started pulling up all the research and saying, these tests are terrible, right? It's likely that he could have had a false negative and that might be what's going on. And so he convinced me to revisit the diagnosis of tick-borne disease. And when I did, I found out that not only did he have Borrelia burgdorferi, the bacteria causing Lyme disease, but he also had 2 common co-infections, specifically Bartonella henselae and Babesia duncani. And so I had our answers, right? And it felt good, but really bad at the same time that it had taken us that long to get there. And so we went to a Lyme specialist and they dug deeper. And as it turns out, the pathogen smorgasbord that my husband had on board didn't end there. He had multiple infections. He had Candida albicans, parvovirus, Epstein-Barr, herpes simplex virus. All of a sudden, I realized my husband, who doctors were telling me over and over again was normal, had literally a pathogen party of all these jerks going on in his body and his brain. And in addition to that, as they ran other tests, they realized that he had extremely elevated levels of lead and mercury in his blood, also not good for the brain. And he had his markers. He had elevated ferritin levels, low iodine, low copper. Does he have Wilson's disease? All these things these doctors are telling me that all of a sudden I'm figuring out all the things going wrong with my husband, but we had lost so much time, right? This was months after we had already been given a diagnosis of late-stage Alzheimer's disease. But at least we had answers, and infections are treatable, right? So we proceeded down the route of treatment. And to say that it was a roller coaster was an understatement, right? I had days where I thought I was going to get my husband and my family back. And then I had days where I wasn't sure I was going to make it through the day. And he responded well in some ways, but not in others. And he continued to decline from a cognitive perspective. And in May of 2019, I made the very difficult choice of putting him in a resident care facility. I was drowning. I had 2 young children. I was still working. He was a full-time dementia patient causing chaos all around the house, and not the nice kind of dementia patient. He was the angry type of dementia patient, and I was drowning. And so the rest of the journey is not pretty. And this is blank for a reason. I didn't take any pictures. Actually, that's not true. I took one. I took one picture and I immediately deleted it from my phone because I never wanted my children to come across an image of him like that. And those of you that have had firsthand experience with Alzheimer's in the late stages of that disease, it is brutal. He was 116 pounds. He was hunched over. I mean, it took away everything he had built, both mentally and physically, and stripped it away. And so in January of 2022, he passed away. And my kids and I are grateful that he was in our lives, and we remember all of the good times. And even with all of the nonsense, I'm still happy that he was part of our life. And so on the other side of that, well, what do you do? Right? Everything I knew had kind of been turned upside down. My faith in certain things, like our medical system, had also been torn upside down. And again, I'm an engineer, right? I like root causes and I like to do postmortems and all the things to understand what we can learn from this situation. And so I had started journaling as part of my coping technique, and I decided to take those journal entries and turn them into a book. And it was part of my healing process, but it was also part of my sharing our story. Because when I would talk to somebody and I would say Lyme and Alzheimer's in the same sentence, they looked at me like I was crazy. And I needed that to change. I needed people to understand how bad these pathogens and these diseases can get when they're not treated. And then from there, actually the founder for the Center for Lyme Action read my book and liked the way that I dealt with the science. And she said, "Hey, are you interested in another writing project?" Okay. She wanted to take all of the amazing research that's happened with Borrelia in the last decade and turn it into a paper, a summary paper that they could use with Congress to lobby for more funding. Because so much research has occurred, mainly through private funding, to show that these pathogens and Borrelia is linked to complex chronic illnesses. And so they wanted to lobby Congress for more funds to basically say, this is a key area that needs more research funding. And so we wrote the State of Lyme Disease Research to help in that endeavor and to show NIH and Congress that more study is needed in these critical areas. And being an advocate and telling my story helped immensely. And it's why I can be here today and not be literally bawling. But it just wasn't enough, right? I kept going back to this problem. And that first doctor's visit where we got that negative result. And as I dug into the research as part of the CLA paper and as part of my own interest, I realized that there are millions of patients experiencing the same thing, right? People showed pictures of the rash, right? That bullseye rash, that typical rash with the clearing. It only happens in 14% of people, right? Other people get other types of rash, and some people get no rash at all. And that makes a clinical diagnosis nearly impossible. And then the testing that we have, a 10-year study by the Bay Area Lyme Foundation showed that it's only effective in capturing 23% of early Lyme patients. 23%. How is that acceptable that a diagnostic test that's the standard of care misses over 3/4 of the people that are infected, especially when it progresses to such complex chronic conditions? And so I started researching all the different technologies and universities and companies that were looking to solve the problem a different way. Like there has to be a better way. And I came across the research of Dr. Breischwart at NC State and Galaxy. And I really liked the approach the more I looked at it, because the current standard of care is based upon antibody testing, right? And that requires the host immune response to have a certain type of reaction. But as we know, immune responses are all different, right? And each of us is different. Some people have a reaction and some people don't, especially when you're chronically ill. And so Galaxy was focused on solving the difficult question of direct detection. How do you directly detect these pathogens and see what's on board? And there's different solutions for different pathogens, because guess what? Microbiology matters, right? And so we have two discrete tests, the Lyme urine antigen test and then the blood-based co-infection test using digital PCR to basically increase the sensitivity so that you can get at these directly and get the diagnosis of the pathogens on board. With great results, right? Really game-changing, providing better answers to patients of telling them what's on board. But the key that I really love about Galaxy is that we're really building a commercialization platform, right? So here, there's so much fantastic research, but I've been doing startups a while, and I know that a lot of innovation happens at the university, right? But getting that technology to the clinic is way different. There is a huge chasm in between those two. And what we're building at Galaxy for new diagnostics and new technologies is a way and a pathway to get that in the hands of providers and clinicians. Take the research that's built at our company and at university partners, and then launch them as CLIA-validated assays and lab-developed tests. And then take that even further and get them FDA approved so that we can disseminate them broadly into kits and get them in the hands of the LabCorps and Quests of the world. Because at the end of the day, what do we need to do? We need to change the standard of care, right? This technology and new innovative technology needs to be in the hands of everybody. And the way that you do that is by taking it to the mainstream. And so as proud as I am of what we're doing at Galaxy, I took over as CEO in June of last year, and we've got a great team. Dr. Jen Miller is here with me today, and she's the brains of the operation, so I encourage you to talk to her. And as happy as I am, the fact that I know we're going to make a difference in so many patients' lives, when I look back at Russ's case, it's still only a portion of what was going on in his case, right? He had all these other infections and toxins and other things. And so this is critical to really understanding this complex illness. And so back to this picture that I started with. So fortunately, the amazing Nikki Schultek helped me donate Russ's brain to science so we could continue to learn from his case. And Dr. Brian Balin is the recipient of those tissues, and his team here has actually done a fantastic job trying to provide the answers that we couldn't get in life. And they have a poster presentation upstairs afterwards. So I encourage you all to go and look at their findings and see what they've learned. Well, what else have we found out? Well, in addition to all the things that I knew about as a caregiver, there was even more, right? So chlamydia pneumoniae, which wasn't even on my radar screen, but Dr. Balin's been studying for years, they found evidence of that in his tissues. And then that Babesia duncani, diagnosis I had struggled with as a caregiver because he just didn't have any of the signs. He didn't have the symptoms. It didn't seem to fit. But he was diagnosed based upon antibody presentation. And antibodies are notorious for cross-reacting. And when Dr. Breischwart, one of the founders of Galaxy, he got some of the tissues from Dr. Balin, he actually looked and said, well, you know what? What we found using digital PCR, looking for the actual pathogen, was not Babesia duncani. We found Babesia urticoliae, which up until recently was only thought to be in deer, right? So it's a deer species, but guess what? We're finding it's more and more relevant in humans, and it's a different type of Babesia. It's a sequestering Babesia, and that's why it wasn't fitting the symptoms. But it was found in his brain tissues, and as far as I know, this will be hopefully when it gets published, the first published case in association with Alzheimer's disease. So we're still continuing to learn and to figure out, and it gives me so much hope that Russ is a part of that journey. And I thank you guys for the work you've done to help provide the answers. And so being here really brings me so much joy. seeing so many of you working together and bridging silos and thinking differently, right? The current thinking hasn't gotten us to where we need to be. There are still millions of patients suffering from Alzheimer's disease. And as Nikki said, it's way broader. These neuroinflammatory conditions expand way beyond that, right? And there are a lot of common roots and elements. And so I applaud you all for thinking differently. I know that it's not easy. And I know sometimes this feels like you're swimming upstream because we do it at Galaxy as well. But I thank you for your work because it gives me hope. And I encourage you to keep going because patients need better than shit happens. They need better answers than shit happens. And so keep pushing and keep driving. And thank you for the important work that you're doing.
Nicole, thank you so much for, just the incredibly moving and thank you, moving talk. And I have struggling with my words because I was at the verge of tears the whole time you're speaking. And thank you for inspiring everyone to think differently and for working with your team at Galaxy to try to change it, what people have access to as far as testing. With that, it's also my pleasure to introduce Dr. Liz Engler-Girazzi from Tulane. Thank you so much, Liz.
Okay, how's the volume? Good, okay, all right. Good, yeah, my voice carries, so I have to be like real conscious not to yell accidentally, but okay, great. So hi, I'm Liz Engler-Girazzi. I'm from Tulane, and I am so incredibly grateful to be able to speak to you today, to the organizers, to everyone who's in attendance and in support of this really important research area, and to my collaborator and partner in this project, Dr. Kevin Swasderek, for losing the coin toss and allowing me to be the one to talk to you today. But hopefully he'll get a chance next year. Okay, so again, I'm so happy not to have to spend a lot of time on this slide convincing you, like I do in so many presentations and on so many grant applications, the importance of studying the effects of infection across the lifespan and CNS manifestations. So I'm just gonna blow right past this and just highlight, as we all know, there's a critical need to understand, characterize, and address the long-term brain health consequences of exposure to pathogens. So right when I got to Tulane, it was the thick of the pandemic. And so the sweet irony of working on this project with Kevin is not lost on me because our first interaction was over Zoom. And we spent the first 6 months of our collaboration never having met in person without a mask or outside. So the fact that we were able to build something, I think, so robust and comprehensive, I'm incredibly proud to be able to talk to you about it. So our model leverages a number of different pathogens, and we'll talk about some of those in a little bit. It exposes these pathogens in a number of animal models, and then we collect tissues and assessments at various time points across the lifespan. Some of those time points are close to the time of initial infection, then some are a little more distal, and then some are after repeat exposures intermittently every 3 months until late in life. Throughout that time, we take a lot of tissues and measures, and then at terminal collection, we are biobanking pretty much everything that we can. And so what I want to highlight is this project is highly amenable to collaboration. So if you see a tissue on this list that excites you or that you're interested, and I think more importantly, if you don't, but you want to see a tissue on this list, definitely get with us because we can collect it, we can package it up and send it your way. We did a lot of work to characterize this model, and we want to make it easier for those in this group to expand upon it. One of the other really cool things about this approach is how comprehensive it is, and you'll see a little bit more of that in just a little bit. But we are collecting, like for example, in the brain, tissues and using them not only for protein level assays, but also for epigenetic and transcriptomic measures, and then correlating all of this out to a behaving animal in a BSL-2 condition. And so what this comprehensive approach allows us to explore is not only what is the cell actually doing, but what it's trying to do, what it's capable of doing, and what is the behavioral output at the end of all of that. So my background in training is in rodent functional assessments, and so we have set up a shared behavior facility within the biosafety level 2+ facility. It's immediately adjacent to the animal housing room. And so they don't have to travel very far. Risk of contamination is very low. And we have a number of tests designed to capture multiple domains of functional performance within an organism. Future tests, we're going to expand into some other models of learning and memory. And then aspirational, one day I really hope to have the very fancy home cage cognitive behavioral monitoring boxes. Those are $50,000 each, so we need a grant in order to do that. But we're real hopeful that we can bring those on board in the next few years. Okay, so just to kind of show you, some of the things that we've been working on over the last few years, I'm going to focus on the brain, my favorite organ. We're going to focus on cytomegalovirus, and we're going to focus on an animal model of human-like Alzheimer's. And then for the sake of time and simplicity, we're just going to look at animals that were exposed to a single infection either a week ago or about 2 months ago. Okay, so why cytomegalovirus? It's a really common virus, right? Most everybody in this room, has or will be exposed to this at some point in their lifespan. Primarily, infection occurs in childhood. And there's a robust immune response to that initial exposure. The virus is never fully cleared, but it is put into latency with periodic reactivation across the lifespan that breaks, with social determinants of health, so inequitably across the population. Now, what do we know about CMV in the brain? Most of what we know relates to the neonatal, gestational, or childhood exposure. And that's, it tends to be really bad, right? There's developmental delays, there can be hearing loss, and a number of other negative consequences for the young organism. But usually this involves encephalitis and brain infection. The understanding of what's going on with CMV in an immunocompetent adult is less well studied and less robust. It's mixed in the literature, especially as it relates to dementia later in life. And so we want to expand upon this literature. The other thing that we like about this virus is that in our hands, in immunocompetent organisms, we think that this pathogen is largely restricted to the periphery. Animals that we're looking at are relatively healthy otherwise, unless they have some layered on transgenic dementia risk. But generally, animals are being exposed when they are young adult and healthy. The neat thing about this model then is that this infection experience and the robust immune and inflammatory response that it induces not only can inform what's going on with CMV, but it can inform also our understanding of intermittent inflammatory and immune activating experiences across the lifespan. So, you go on vacation for a couple weeks and eat cheeseburgers every day. Is that having CNS manifestations that persist into the long term? So this has implications, if you permit me to reach just a little bit, well beyond pathogens, but more in that exposome domain, which I know is such a great hot topic. If this sounds like a sales pitch, it is. I'm going to have 2 R01s under review in the next 6 months addressing various aspects of this topic. And I am currently accepting applications to write tenure and promotion letters on my behalf. So if you like what you hear, definitely reach out. Okay, so right now we are starting with the triple transgenic Alzheimer's disease mouse model. This was a classic Alzheimer's model developed by Frank LaFurla in the early 2000s, and we like it for a number of reasons. There are 3 different transgenes, humanized Alzheimer's-related transgenes that are represented in this animal that capture both A beta and neurofibrillary tangle pathology. So we're trying to capture the spectrum of the classic hallmarks. There's also evidence of neuroinflammation, LTP disruptions, and cognitive deficits. And because this model is so well characterized in the field, using it allows us to back translate really effectively and layer on what is known from the literature, what is known now about the infection paradigms that we're implementing that are a little bit novel and different onto the historic literature. Okay. Now, everyone has their favorite model. We are starting here. And we like it in part because it's relatively robust and relatively fast progressing. There are faster models. And so, it just really depends on the question that you're asking. We like it because we can still have wiggle room to accelerate the emergence of certain pathologies or exacerbate others that are already present at the time points that we're measuring. Okay, I probably also don't need to spend a lot of time selling this group on the incredible, amazing complexity that the brain brings when you're trying to study it. I mean, it's probably what fascinated me most about the brain when I was a student was seeing just how dynamic it can be in a matter of microns. And so while interesting, that complexity means that we need to really deploy cutting edge techniques to understand, sort of region by region, what the effects of exposure to a pathogen might be doing. And to do this, we need some cutting edge tools. Tools I never, as a psychology undergraduate, thought I would be using. I thought a Western blot was fancy. So the fact that we're doing these amazing transcriptional and epigenetic readouts is really, to me, quite incredible. So we're going to take you through the next couple of slides, our validation of using spatial multiomics and integrating this information across animals. So the first step in making sure that our framework is working is to show that we can reproducibly collect the brain for all of this processing. So here are sagittal sections from 4 different subjects, 2 mock on the far side of the screen. That's going to be your right. And the other 2 are infection experienced, at some point in the past. And I want you to take away from this slide that within a matter of probably about 50 microns plus or minus of this 10 micron slice, we're like right in the same range. So this allows us, this sectioning technique that we've developed over months it took to get right, fairly consistent regions of interest from subject to subject that only really differ on their experience with a pathogen. Okay, the next part of this optimization and validation model was to make sure that our 10x multiome data is representative of the distribution of cell types within the brain, as is our 10x xenium data. Now, I can take almost no credit for this, but who I will point to is Stella in the corner. This is a very enterprising and hardworking graduate student in Kevin's lab. I have the privilege of getting to be on the committee and watching this brilliance unfold. She has worked really hard to validate the integration of these different data sets. So if you have questions, I will be happy to try and answer them, but I will go get the real answer from her. Okay, so what this data is showing is the top figure is our multi-ohm data mapped onto the Allen Brain Atlas, and the bottom is our xenium data, the spatial data, mapped onto a sagittal section, also from the Allen Brain Institute. And what we're seeing is we have really nice correlation and agreement between our data sets. So we have high confidence that our data is providing a reliable representation of the cell types in the nervous system. Okay, so we sort of checked off that we are in agreement with commonly used and accepted tools within the field. We'll come back to that in a little bit. Now we want to try and make sure that we can integrate our multiome data with our Xenium spatial transcriptomics data. So on let's see, the first image here on I don't know what side, I can't tell the difference between my right and left hand, so I think your left. So these are Xenium data clusters that we collected from a particular sample. It's supervised clustering, so we only have 15, but we can also do unsupervised, which generates usually about 50 unique clusters of cell populations that are transcriptionally similar. And then in the other figure, we've overlaid our multiome data that each little black dot reflects a cell from the multiome that is transcriptionally similar to a cell that is present in our Xenium spatial data. Okay. Demonstrating that these have similar cell profiles. From there, we can start to ask questions about where these cell types are on the Xenium slide, and then link that back to the cell populations from the multiome that have a similar transcriptional phenotype. And so when we do that, the green dots are sort of all cells that we were looking at. The red are cells with pretty high confidence that they are the same transcriptional profile. And then what we can do is then subtype them. And we found, for example, these olfactory bulb GABAergic cells, really great alignment from the cells that were predicted to be transcriptionally these cells with where they were actually showing up in brain, which is the olfactory bulb right there, in case anyone is not familiar. This is just another validation step, again, sort of taking our spatial multiome data and pairing it back to the MERFISH, which is another sequencing approach that uses sort of different machinery in order to generate the transcriptional profiles. And what this graph is showing is that we get pretty good alignment between our cell types, transcriptional profiles, and those of MERFISH when we apply that approach and tool to the data. Pretty good. It could be better. I'll give you that. That said, I should point out that our animals were differing from the MERFISH data set because they were a different sex, they were slightly different in age, and they were on the 3xTG transgenic background. The animals in the MERFISH were not. This is yet another way of showing that we have pretty high confidence in the agreement between the MERFISH standard accepted data set and ours. What this is showing is that we have a number of cells, a reasonable number that have very high confidence of alignment. As we increase the confidence level, as expected, the number of cells present in that map is going to go down. But we're pretty pleased so far with the amount of alignment that we are getting, and we do have plans to follow up with animals that are nearly identical to those from the MERFISH, atlas, for lack of a better term, and making sure that our approach with our spatial multiome is mapping on as closely as we can possibly do. Okay, so now that we've sort of validated the alignment between our different approaches, we can start to ask some questions about what's going on with infection. So that's really exciting. We are at the incipient stages of this, but just as a brief spotlight, we've got these really interesting ITET, intratelocephalic and extratelocephalic glutamatergic neurons as an example. This slide is showing that when we look at these clusters, we get sort of 3 different clusters of these same neuron populations. They each have slightly different transcriptional profiles, but what you can see is that they're mapping on really nicely to the hippocampus within the spatial representation. So we can begin to ask pointed questions between infection experience states, which we're excited about. When we look at some animals that were exposed to cytomegalovirus at 2 months of age, 2 months ago. We find interesting changes in various pathways implicated in a number of neurodegenerative diseases, including Alzheimer's, to say the least. We see lots of genes that are differentially expressed, and the likelihood that they are due to chance is extremely low. So we are excited to dig into specifically what those genes are and to manipulate those in the future. Okay, I gotta pick the pace up here a little bit. So I am old enough to remember at Society for Neuroscience when it was all neurons all day, all the time, and that's all we ever wanted to talk about. And so, we got to look at microglia because those have really, risen to the forefront as important cells regulating all manner of things that the neurons need and do. And so when we look at microglial subsets, we see really different patterns of expression between an animal that was previously infected and a mock animal, including the presence of populations like 8 there in the bottom right of our cluster figure that's completely absent in an animal that hasn't had an infection experience. It's also, we're showing when you look at specific genes that might be changed. We see upregulation in genes implicated in cell activation, antigen presentation, amyloidosis, and neuroinflammation, among many others. Okay, so sort of our takeaway from this right now is these cells are different. We don't know if this is good or bad, but they have been perturbed. They are shifted away from a homeostatic phenotype. They're not yet normalized, and we don't know why. But interestingly, the microglia even aren't the cells that are changing just in isolation. We're seeing remarkable increases in the proportion of both astrocytes and oligodendrocytes. And when we take a look specifically at the oligodendrocytes and the genes that have been significantly altered, we see changes in cell activation and activity-dependent transcription. So I can tell you, I definitely go to SFN and don't see a ton of talks on oligodendrocytes. So hopefully in a couple of years, we'll be one of those people, exciting people about these cells as well with data like this. Okay, this is integrating the expression, the transcriptional expression with our ATAC-seq. So at 1 week post-infection, which is the top 3 graphs, in astrocytes, oligodendrocytes, and microglia, we can see the blue dots representing areas of open chromatin accessibility. And it really does seem like the astrocytes and the oligodendrocytes are the ones that are showing the most regions that are open. When we look 2 months later, again, here we see that in all 3 of these cell types, but the astrocytes and the oligodendrocytes, I think more so than even the microglia, these transcriptional profiles are still shifted and they have not yet returned to normal. I'll skip this for now. Okay, so future directions, really quickly, where are we going? First, we're going to take over the world. And on our way to doing that, we are going to develop a method to convey this data from this fully integrated model. We haven't yet figured out how to display all this data. So if anyone has ideas, we are open to suggestions because these are massive data sets and, sort of like a 3D figure that shows the ATAC-seq, the behavior, the protein, and the transcriptional changes in a single gene at a single time point. I don't really know how to do this yet. So we're on the path to figure this out. Another really cool thing that the spatial is capable of doing, and it's something that we're proposing to do in a number of proposals where several folks in this room, Will, Brian, David, who's not able to be here, David Corey, is to look at various pathogens and to ask the question, what is the transcriptional profile of an infected cell within the brain? Some pathogens we know are neurotropic, so they definitely can get in, they definitely can infect cells, especially those within the hippocampus seem to be particularly vulnerable, and we can get a sense of what their transcriptional state is. But then we can compare that to an infected cell of a different cell type that's nearby. We can also compare that to a non-infected proximal neighbor, a non-infected distal neighbor, or a non-infected cell in a completely different brain region, and ask the question, what is going on with infection experience? And then lastly, we can disseminate this. So that's what we're doing here. But I also want to highlight something that's really become very important is highlighting for the community what we're doing, because we exist now in an era of anti-science rhetoric. And I never thought as a student studying Alzheimer's disease that my research would be considered controversial. And yet, here we are. So a big part of my job, in addition to working at the bench, is to communicate science with the public. Kevin has been a great partner in this process. So you can see we have a number of events that we attend where we engage community members learning about the brain, talking about neurological diseases, and also trying to recruit the next generation of scientists. So I'm over time. Super thanks to everybody who's been a part of this research program, and please call us. Thank you.
So much. Thank you, Liz. So, unfortunately, due to the government shutdown, Michael Duggan, who really wants to be here with us in person, is not able to be here, but he is going to present to us here on Zoom, thankfully. Dr. Michael Duggan is a member also, like Juan Fernandez, of our AlzPI Next Gen group, and we're very excited to have him join us here today on Zoom. Michael, are you hearing us okay? And
Yes, I can. Yeah, can you hear me all right?
yes.
All right. Can everybody see my slides? All right. I'm not hearing any nays, so I'll take that as a yes. Yeah, as Nikki mentioned, unfortunately, I'm unable to travel. I apologize he can't be there with you. I was really looking forward to this for quite some time, so thank you very much for accommodating me. I have a timer going, so I'll try my best to keep on time, but if I go over, just holler at me. So yeah, my name is Michael. I am speaking today in my own personal capacities. I'm not affiliated. I have to say I'm not affiliated with anybody. And yeah, I'm excited to talk to you about a project recently that came out of the lab where we identified what we think might be some key peripheral proteomic drivers of some of the post-infection consequences we see on brain structure and function. So the crux of the data that I'll be presenting today really focused on using infections and microbial exposures as more or less a probe to understand the biological mechanisms that might be contributing to neurodegeneration and dementia. We all know that dementia is an extremely heterogeneous condition. The biological mechanisms are equally variable, and we also know that immune mechanisms play a primary role in the incidence and prevalence of some of these conditions. However, we actually don't understand to at least the greatest ability that we could, how some of these viral, bacterial, and other microbial exposures impact your brain structure. We do know that there is some evidence, at least in acute severe settings, that COVID-2, as well as HIV, can influence and have deleterious effects on brain structure, but we actually don't know whether or not these effects persist over time. In the Baltimore longitudinal study of aging, or the BLSA, we have shown previously that individuals who have a history of herpetic infections do experience accelerated rates of brain atrophy, particularly in the temporal lobe, and some of the other white matter regions of the brain. However, other data coming out of the Diane cohort, so these are individuals who express familial AD mutations, there is some evidence there that, at least with regards to whole brain atrophy, individuals with herpetic viruses don't display any accelerated rates of brain volume loss. Accompanying this lack of knowledge, we also don't understand, at least to the greatest degree, how these pathogenic exposures influence our peripheral immune proteome. And here I'm referring to the array of proteins that are expressed by peripheral immune tissues and effector cell types. And the reason why this is relevant is because we know through a variety of complementary mechanisms, it's these proteomic mediators which can influence a variety of neurodegenerative mechanisms in the brain. And as I said, through a variety of complementary mechanisms, not only interacting with cells along the blood-brain barrier, but also direct interactions with CNS cell types. So perhaps, not surprisingly, we also see that some of these peripheral immunological mediators have been previously linked to differential effects on brain structure and brain health, at least in regards to singleplex assays in which they were measured. Moreover, extending this research question a little bit farther, might there also be some of these peripheral proteomic mediators which are not only differentially expressed among individuals with a history of different infections, but might they also be capable of influencing changes in brain structure which are themselves susceptible to infection-specific atrophy? To answer this research question, we used our discovery cohort of the Baltimore Longitudinal Study of Aging. Individuals here are seen biannually if they are between the ages of 60 and 80, every 4 years if they're under the age of 60, and annually if they're over the age of 80. Follow-up here, at least for baseline, began in 2009, 2010, when all of these individuals, approximately 1,000 that were in the study, had both their baseline MRI scan as well as a blood draw for subsequent proteomic measurement. Individuals were considered unexposed or negative cases if they lacked any history in their medical diagnostic reports of an infection-related diagnostic code, whereas individuals who were considered positive cases or exposed here, their earliest exposure date was set when the presence of an infection-related diagnostic code was detected. In addition to asking how these medical diagnostic infection-related codes relate to longitudinal changes in brain structure, we also use these same infection categorizations to ask how that might they be related to differential dementia risk in the UKB over approximately a 14-year follow-up period, and in a Finnish multi-cohort sample where the follow-up period was about 20 years, 19.7 actually. Moreover, we also asked how these same infections influence the peripheral immune proteome, and here we used the SomaScan assay, particularly their immune and inflammation response panel, which consisted of about 940 proteins derived from their larger 7K platform, which measures the abundance of approximately 7,000 proteins. We also went on to identify candidate proteins, which, as I mentioned previously, we defined as not only differentially expressed among individuals with a history of certain infections, but also are these proteins which are differentially abundant among these individuals, might they be capable of predicting changes or rates of brain atrophy in those regions of interest which are themselves vulnerable to infection-specific brain volume loss. Having identified candidate proteins, we also conducted a variety of complementary supplementary analyses to characterize these proteins with regards to cross-sectional and longitudinal cognitive functioning, and also using a multi-cohort approach, how these candidate proteins might be related to canonical ADRD biomarkers. Finally, we provided a variety of bioinformatic analyses, which further drilled down on the specific biological pathways through which our candidate proteins might be influencing brain structure and function. In total, for our MRI analyses, we had approximately 980 individuals. The follow-up time, as you see here, was about a little bit over 5 years. In total, we identified 15 different infection categories. Some of the specific infections, for example, miscellaneous viral infections and URTIs, upper respiratory tract infections, are listed below. We had over half of our individuals, our participants, having a history of any sort of infection, where only about 10% had a history of more than one infection. And I would just like to note, these electronic health records were primarily derived from GP diagnoses. So they're relatively subliminal exposures compared to more severe or acute hospitalized infection exposures. And along with that, we identified a mean time of about 16 years between your first documented history of an infection and your baseline MRI scan. So these are really proximal exposures that we're going to be discussing here today. For 6 of our infections, we actually identified accelerated rates of brain atrophy in multiple different regions of interest. influenza, herpetic viruses, miscellaneous viral infections, upper and lower respiratory tract infections, as well as skin and subcutaneous infections. We also conducted cross-sectional analyses where we didn't identify any differences associated with these specific infections, suggesting that these longitudinal accelerated rates of brain volume loss aren't being attributed simply due to a regression to the mean. We also didn't observe any dose-dependent effect, meaning if you have more than one infection, do you see accelerated rates or proportionally accelerated rates of brain volume loss, suggesting to us that these effects are somewhat specific to the specific infection categories identified here. And moreover, we then, as I mentioned, took these exact same medical diagnostic codes and across two different large cohorts, both the UK Biobank and in the Finnish multi-cohort sample, we identified elevated risk of dementia associated with each of these in one of these two cohorts. And what I hope you can appreciate here is that while we did observe elevated risk of dementia for all cause, as well as Alzheimer's disease dementia, what we really observed was a disproportionate elevated risk of vascular diagnosed dementia, which is something that is supported by some of the recent data as well. We then asked, how do these same infections, which are associated with changes in brain structure, associated with variation in your peripheral immune proteome? And the largest proteomic signature, surprisingly, was associated with influenza, where we identified 141 proteins which were differentially abundant among individuals with influenza history. And the first thing we did to assess the validity and reliability of these proteomic signatures was just to look at the individual proteins and seeing if there was any prior evidence that suggested their relevance to the given infections. And indeed, we saw things like neprilysin, a prominent amyloid beta degrading enzyme, being downregulated among individuals with influenza. Higher levels of TREM2, a key mediator of AD pathophysiology, being elevated with human herpetic viruses. Also, elevated levels of IFIT3, one of the most well-known antimicrobial peptides with upper respiratory tract infections. And other things like elevated levels of ADAM17 with skin and subcutaneous infections, which is a well-documented TREM2 sheddase. In total, as indicated by the proteins labeled in gray, we identified 32 proteins which were associated with one or more infections. And again, based on existing literature, these suggested that we're indeed picking up on some valid proteomic signatures associated with these infection exposures. Things like RAG1, one of the most well-known VDG recombination activators. Also, another key antimicrobial peptide we detected, DEF-P4A. And we also saw variation in multiple different isoforms of apolipoproteins. Furthermore, again, to build some confidence in these proteomic signatures, we searched through the literature to find any other proteomic studies that examine changes in the proteome as a result of any of these different exposures. We surprisingly didn't find very much. We did find a variety of in vitro studies that examine changes in the proteome associated with multiple different viral exposures. And when we took a look at these studies, the vast majority of them, unfortunately, were from an older proteomic platform, the SomaScan version 1. But again, here we observed quite a robust overlap between the proteins that were measured on that platform and the proteins detected in our analyses as being differentially abundant among individuals with these prior infections. We then went on to identify candidate proteins. So again, these are proteins which are not only differentially abundant among individuals with a specific infection, but they also predict longitudinal rates of brain volume loss in those regions which are themselves susceptible to infection-specific atrophy. And what we observed was a clear population or a higher abundance of proteins which were seemingly pathogenic, and perhaps not surprisingly, lower levels or lower abundance of proteins that were seemingly protective. So, for example, if we take a look at influenza and we hone in on a protein called PIK3CG, what we see is that this protein is lower among individuals with a history of influenza, and by itself, it's seemingly protective, and in this case, it's protective for temporal gray matter. And the reason why that's relevant is because temporal gray matter in our discovery analyses came up as significantly accelerated loss among individuals with influenza. So again, we saw this pattern repeated over and over again, higher levels of pathogenic proteins and lower levels of protective proteins among individuals with these different infections. So to gain further understanding with regards to what biological or pathophysiological relevance or roles these proteins may be playing, as I said, we examined a variety of different AD-related endophenotypes, not only cross-sectional, but also longitudinal rates of cognitive functioning, and also canonical ADRD biomarkers. Again, this was a multi-cohort approach, so we leveraged data from the BLSA, Generation Scotland study, as well as ERIC. And while we did observe some pathogenic associations with our deleterious proteins, as indicated on the right-hand side of your screen, what I hope you can appreciate is that we observe a clear pattern of association with regards to our protective proteins. So again, these are proteins which are lower among individuals with a history of different infections, but with regards to brain structure, cognitive functioning, and ADRD biomarkers, they seemingly exerted protective effects, suggesting to us that the higher levels of pathogenic proteins might not necessarily be so important for mediating the associations with these infection exposures, but really this might be a story of vulnerability or susceptibility where these individuals have lower levels of otherwise protective proteins. And it's this lack of protective proteins that is ultimately leading to deleterious effects on brain structure and functioning longitudinally. We also conducted Mendelian randomization analyses. So this is a unique causal inference technique that takes advantage of genetic information, naming specifically proteomic or protein quantitative trait loci that influence changes in our plasma proteins. And we also leveraged a genome-wide association study of brain volume loss. And this allows us to infer causality, and we observed a variety of different associations here, much more than we were expecting. So again, if we focus in on PIK3CG, what these analyses tell us is that the genetic underpinnings driving PIK3CG levels in plasma on the SomaScan platform are actually causally related to preserved brain structure, at least with regards to total brain volume over time. Again, suggesting to us that these mediators might not only be associated with some of these deleterious effects of infections, but they actually like be causally and mechanistically relevant as well. I don't have time right now. I'm looking at the clock. I'm at 15 minutes to dive into some of the specific biological pathways that we identified. But with regards to the cell and tissue specificity of these candidate proteins, which we think might be particularly important in driving some of these effects, and paying attention to these pink rhombi, I think rhombuses, pink rhombuses. What I hope you can appreciate here is that most of these proteins were not emanating from CNS cell types, which suggested to us that a lot of these effects might indeed be mediated by proteins emanating from peripheral immune tissues and cell types, which are then exerting their effects on brain structure and function longitudinally. So just as a quick summary, we identified a variety of different pathogenic or microbial exposures, which are associated with not only elevated dementia risk, particularly vascular dementia, but also accelerated rates of brain volume loss, most prominently in the temporal lobe. We also characterized the proteomic signatures of these infections of interest, and moreover, we identified candidate proteins which might be mechanistically relevant to driving some of these effects, where we identified that there might not necessarily be higher levels of deleterious proteins, but it's the lower levels of seemingly protective proteins which might be primarily contributing to the consequences of these pathogenic exposures. I'm not allowed to say who is and who is not involved in this study. I can just say that we're thankful to everybody who was involved. I can say that, we appreciate and acknowledge the participants. And I can just say that this paper was published. So if you have any additional questions, please feel free to reach out to me or refer to the paper itself, which has been print now. And again, thank you very much for accommodating me today and listening and for your attention. Thank you.
Thank you. So our next speaker is going to be Tomas Philipp from the University of Sherbrooke. Welcome, Tomas.
So good afternoon. Thank you very much for inviting me and giving me this opportunity to speak here. And thank you for Nikki to put together such a wonderful symposium and also giving us the possibility to networking. So thank you. So I am from Canada, Sherbrooke, and I am from Quebec, and I am a geriatrician. So I see patients, and after what we heard here, never ever for the 40 years that I made geriatrics, any of the patients asked for infection or any other causes. But recently, everybody is asking about the monoclonal treatment and how they can get it. So which means that, unfortunately, at the patient level, this concept of infection doesn't exist. So we should do perhaps more work on that at the patient level. I don't know. I try to convince them that vaccination and other things are very important, but this is just one thing. Here, we had a study, we recruited about 100 persons with different stages of Alzheimer's disease, and we were interested in parodontitis, as we had our colleague from McGill, Dr. Nguyen, who is a dentist, a parodontist, who came to examine all the patients that we had recruited. So here is one part of the study, and this is the circulating extracellular vesicles containing P. gingivalis outer membrane vesicles from periodontitis. Patients may penetrate in the brain and trigger the release of inflammatory markers and the amyloid beta peptide inducing neuroinflammation. So these are all the persons who I think who participated. You have their name here. So periodontitis is a chronic inflammatory disease, as you know, and its key pathogen, Porphyromonas gingivalis, has been implicated in Alzheimer's disease, also in rheumatoid arthritis and many other chronic inflammatory diseases. But still, this is still debated. So there are many publications, but time to time, there is a publication which tries to negate everything that it was done in this field. And the underlying mechanism linking these oral pathogens to neurodegenerative pathology remains still poorly understood, because most of the people had many difficulties to see how the mouse can be related to the brain and how, after having for 10, 20 years this pathogen in our mouse, can lead to the Alzheimer's disease or any neurodegenerative diseases. Okay, so there is some missing, but nevertheless. So this is the third slide in that. So which shows that, you have different ways for this pathogen to go and increase the neurodegeneration. One is the direct way, even if in most of the brains that were examined, they could never show that the Porphyromonas gingivalis is present. So this is about what we have heard in the stories today, that it's very difficult sometimes to show that the viruses or bacteria can be directly in the brain, except some that other colleagues like Brian has shown for chlamydia. But, and also there are indirect ways, and these indirect ways can be somehow more important than the direct invasion. And one route is what we call the outer membrane vesicles. The outer membrane vesicles are derived from these bacteria, which are always just shedded from them, and can contain a lot of components of this bacteria, mainly LPS and others. So you can see here how it can be complex from one bacteria to invade and cause neurodegeneration. So this is what we have, but this is just part of the patients that we try to, by shotgun, measure the presence in the saliva Porphyromonas gingivalis and Treponema denticuli, which were involved in neurodegeneration. And you can see that almost all the subjective memory patients have Porphyromonas gingivalis, and many of the stages of the Alzheimer's disease has more frequent presence of these bacteria. But in the early stages, there are overwhelming presence of Porphyromonas gingivalis. This is we try to see in various cells of the organism, and mainly in the immune cells, whether you can find with the cells RNA that we have heard this morning, how it is. And in the exosomes or extracellular vesicles, we could demonstrate the presence of bacteria in mild cognitive impairment and Alzheimer's patients. So the study aims to investigate the systemic dissemination of the Porphyromonas gingivalis and its contribution to Alzheimer's disease pathology through the outer membrane vesicles. Specifically, we will identify the Porphyromonas gingivalis-derived OMVs in the plasma of periodontitis patients and assess their impact on microglia and neuronal cells. You know, the extracellular vesicles are very difficult to separate. There are at least 100 different methods to try it. And now we have flow cytometers that you can use to separate these, because they are very, very small one. And it's not enough to see them under the microscope, but you should also characterize them. You have here what we have from saliva and also from the plasma. And here we try to characterize them. And you see here that the most important markers, I don't go to the details, but these are characterizing these extracellular vesicles, and the negative marker is connexin. So none of them had, so we are very sure that what we have are really extracellular vesicles. And also, we try to see whether inside of these extracellular vesicles, we can trace the proteins of Porphyromonas gingivalis, and you can see here the gingipain, and this is here the gingipain alone, and you can see here in the saliva and also in the OMVs, in the saliva and plasma EVs, that the gingipain is present. Another marker, which is the GRUEL protein, is a stimulator of inflammatory cytokines in macrophages. So we are pretty sure that in the extracellular vesicles that we have separated, we have the presence of OMVs, which are derived from Porphyromonas gingivalis. And what is interesting that when we try to treat HMC3, which are human microglial cell lines, and also this neuronal Swedish mutation-containing cells, they don't kill the cells. It's very important because many persons relate these infections to the decrease of neurons or any other cells by cytotoxicity, but as HIV, as herpes simplex, they don't kill these cells because they would like to survive. So their interest is to make these cells viable. We saw for the uptake of these saliva or plasma-derived extracellular vesicles by HMC3 or these neuronal Swedish cells, and you can see that the microglia are actively taking up these extracellular vesicles and also the OMVs that we have obtained from a special culture of Porphyromonas gingivalis. This is not the case for these neuronal cells. And the next is the production of inflammatory cytokines. And you can see here various cytokines. And you can see that the PG-derived OMVs and also the saliva and the peripheral plasma OMVs can stimulate these very pro-inflammatory cytokines. But what is interesting, because most of the people never measure the anti-inflammatory cytokines, which is IL-10 and IL-4. This is always a yin-yang between the inflammation and the anti-inflammation. If you have inflammation, you have straight the beginning of an anti-inflammatory response. So the question is how it is balanced at the outside of the cells, what are the most important, and how they can increase or decrease the inflammation. But what is interesting that they can really increase this inflammatory and anti-inflammatory process. Here we try to use another cell line, which are the THP1 monocytes, which are not the microglia, but just monocytes, and compare of the OMVs from P. gingivalis and E. coli, because the question is whether all bacteria can do the same, or this is very specific for Porphyromonas gingivalis. And you can see somehow they are identical, but somehow they are very big differences. For example, for IL-1, which is a very important factor and pro-inflammatory molecules in neurodegeneration, the P. gingivalis is much more important than E. coli. So it means that even if they are all the same genus of bacteria, they don't have the same effect on the inflammation of monocytes. And this is the production of phospho-tau, and you can see here that both of them in different cells, like the first is human neuronal cells, they can increase the phosphorylation of tau. This is the 231. There are many other types now, 217, 181. So what is this one? And you can see that really it increases, but not significantly different when you use this genetically modified other neuronal cell types. This is the production of reactive oxygen species, and they were also involved in the Alzheimer's disease. And you see here that in the microglia cells, the P. gingivalis OMVs are much more important to generate free radicals than the E. coli. And the other cells are also very different from one to the other. This is when we injected this to the tail of a zebrafish. 3 hours later, it appeared in the brain of this zebrafish. So it means that whatever and wherever is your infection, there are some mediators which can bring these OMVs to the brain in anyhow. And this is another experience that you can hear, that if you apply the beta amyloid to the supernatant, which is the planktonic culture of Porphyromonas, they can kill and decrease this culture, which means that the P. gingivalis is really sensible until they form biofilms. And the strong characteristic is to make biofilms, because when they make biofilms, the beta amyloid become pathogenic, and they will favor the existence and the growth of these biofilms. So the same dose of beta amyloid, when they are at the beginning, is very helpful, but when you are at a later stage, they are very harmful. And you can see here that the A beta is forming, fibrillation and everything. But if you see that with the parts of the biofilm from Porphyromonas gingivalis, they become really sticked and they are not removable at all. And this is here, that if you have the monomeric A beta, the gingipine is able to destroy it. But as soon as you have a biofilm, this is not possible. So which means that until it becomes later, years later, the infection and chronic infection the beta amyloid is at the beginning very helpful, and it can really help to avoid the infection. And this is not shown, but it is shown that P. gingivalis is able to increase the secretion of beta amyloid from neuronal cells. So it is a sort of mechanism which in the meantime tries to avoid, but this is also a mechanism which later become more harmful. So this is just a cartoon to show how it is the neuroinflammation and the OMVs. And so our results show that the circulating EMV contains P. gingivalis OMVs as confirmed by Western blot and transmission electron microscopy and immunogold labeling. The circulating EMVs injected to zebrafish traversed the blood-brain barrier and were found in the brain 3 hours after injection. And the EVs triggered the secretion of inflammatory cytokines by these human microglia cell lines, and also the production of phospho-tau. That the study demonstrated that the plasma-derived EVs from periodontitis patients can trigger inflammation and the production of A-beta, highlight the systemic spreading of these OMVs and the contribution of them to the development of neurodegenerative disorders. And this could be considered as a critical tool to discover systemic markers of neurodegenerative diseases in periodontitis patients and can be somehow sort of druggable target at the end. So thank you very much for your attention. Oh.
We have to change this. We need Yuri's. Thank you. So our next speaker is going to be Yuri Kim from MIT. Welcome.
Hi, everyone. Thank you so much for this wonderful opportunity. My name is Yuri. I work at MIT with Dr. Tal, who presented this morning. I am a research study nurse for the MIT Meister study. I am the main research nurse, has been honored to meet more. We've been actually accumulate just 175 participants, and out of 200, 210 that we're aiming. So I just want to share my insights as I have had this journey as a research nurse meeting all these participants. So Mickey talked a lot about the predictive diagnostics and our mouse experiment. I am in that box doing this neurocognitive testing with our participant. So we're going to talk more about it. Just like the Rubik's Cube that was presented early in the morning, I always think about this as a very complex chronic illnesses associated with the infection. I do often think about this, and I would like to talk more about how complex this gets and how, in my personal experience with the participants, how I learned we should approach this neurocognitive impairment. Meister study, we have a lot of aims here, but I just wanted to point out some of the things that we are really trying to aim. We're trying to aim new biomarkers of the illnesses, and we try to phenotype to help inform future studies and trials. And we are gathering massive data from the current participants. It is an observational study, and we're hoping that from this type of data, and we are analyzed very detailed and subgroup of a participant with a different phenotype, and we want to later on perhaps do more complex interventional study that can be potentially therapeutic to a lot of participants to help out this community. This is the kind of approach, the field that we're trying to really investigate further, from allergic responses to gynecological problem and menstruation. I'm hearing a lot of stories from our female participants, particularly who's really going through a tough time with these neurocognitive changes. And the female participant we're recruiting from age 18 to 65. And I have learned, and this is just purely my insight, but there is a greater play interconnection with the hormonal changes in women's lifespan too. So that's something we also try to understand better. And mitochondria function, immune dysregulation, and microbiome, blood flow, vascular dynamics. I'm also realizing that blood flow and vascular structure is a lot to do with the neurocognitive impairment. And how can we interconnect all these dots together? That's what we try to do. So I'll particularly talk about the end box here, neuroinflammation, neurocognitive, blood-brain barrier dysfunction. As Miki suggested, we want to measure absolutely everything. So I'll go more detail about our study here. Maestro study, currently, we're aiming to recruit 240 participants, which is 60 per cohort. Acute Lyme, those people will have the bullseye or the tick bite observed or diagnosis within the 4 weeks before they visit here. And then the chronic Lyme and long COVID, we have participants who have suffered from these lingering symptoms for more than a year. Our Lyme participants should have tried antibiotic treatment upon initial diagnosis, and the symptoms still linger after a year. And control group, we have an interesting control group, people who have diagnosed with the Lyme disease and recover, and then long COVID and recover. They got sometimes both and recover. So those are the control group who do not have any functional impairments. This is a kind of good picture of how we design our study, and I would like to emphasize the importance of collecting the data from patients. We use a lot of validated survey. Study visit takes about 4 to 6 hours, and it requires a person to come one day study visit site. But it just really exhausts our participant, and regardless, they're so willing to provide as much information as they can throughout the very thorough detailed survey that we provide and ask. And neurocognitive testing, we do a lot of testing here. I'm going to particularly talk about the eye tracking testing and the EEG testing that we run, and so battery testing. And also we do a lot of multiple biological sampling, which we recently did a very early analysis about 2 days ago, and I will briefly mention that in my talk later on. What I find the most important part when we collect the research data and to be a great foundation to develop any therapeutic target in the future is patient information and their narratives, their journey. I really just try to take as many notes as I can, but this is the key information that I have been able to understand better about the complexity and heterogeneous symptoms of the brain fog. And I just wanted to emphasize that part. So I will dive deeply more into the neuroinflammation and neurocognitive blood-brain barrier dysfunction here. We ask a classic survey of the PROMIS short form cognitive function, those 8-item questions asking about one's cognitive function. And I want to also talk about the BIVSS is a long acronym, but Brain Injury Visual Symptoms Survey. I found this particularly interesting because a lot of our participants have complained of severe sensitivities to light and severe visual disturbances that developed over time that is absolutely associated with their cognitive function too. I personally think that this particular survey is very vital to correlate with the eye tracking testing that we do here. And then we also ask about individual functional capacity, fatigue scale survey, and then again, patient narratives to really ask, when you were experiencing this issue, how were you back before you got this infectious disease? Each time point and new onset of symptom, I just have to dig further about the time set to understand this illness trajectory better. With that said, we are currently collaborating actively with the Columbian University. Mara Coverdina is working at the Columbia University there. So we're going to dive deeply more into the analysis of EEG data and all this data that we have accumulated. Okay, so brain fog. that's a common term that patient describe, but some anecdote that I want to share from my journey as a research nurse is this is a very subjective symptom that a lot of our participants have tried multiple clinics to address this issue. All our participant wants to do is just simple. They just want to have their life back where they were before. They were loved ones of their a family and children. It's a mother of 3 to 4 kids. Just cry in front of the study room because all they want to do, they're just so devastated that they see themselves not functioning as a mom or as a spouse, or they get divorced because the spouse wouldn't believe this symptom, and sometimes being told that this is all made up. So it is reality of these participants that I'm honored to hear their story, but it is absolutely the devastating problem that we need to try to understand and how to approach this better. In our study, brain fog, we just asked what is the currently most debilitating symptom that affects your quality of life. In chronic Lyme, we have about 58 participants, and 98% in that cohort complains that brain fog is the most debilitating symptom. Long COVID is 95%, and control group, it's 6%. So it is clearly the big problem here. How would people describe brain fog? Because from what we understand, it's oftentimes long-term memory loss or concentration issue, and a lot of difficult decision making. That's what's commonly known. But I try to understand better about subjective symptoms that every individual describe, and sometimes this is about difficulty processing sound. Difficulty processing the visual sensitivities and visual stimulus. So I find it personally interesting that how people describe this into detail. And I wonder some of the neurocognitive testing that we're applying in our study may have the correlation with this patient-reported outcome further. So that'll be another analysis we're approaching here. But I also want to share this one literature that I particularly like. Meg Werther just basically dissecting the whole description of how people will describe the brain fog. I would like to have all of us to look at the pink circle here, because this is sometimes what I often hear from study participants. It's derealization or dissociation. It's almost like the body and mind won't engage each other. It's so surreal for these people to experience this after infectious disease because they were highly functioning professionals before they got sick. So this description matters, and I'm trying to also really see how we can better develop specific surveys that really distinguishes this subtype of the symptom complaints. Okay, I'll probably briefly mention this. Promise, this is all 8 items here. But as you can see, so this is one of the symptom burden that we're suggesting here. The green box is the control group, and long COVID and chronic Lyme clearly have the greater symptom burden. In this particular survey, higher the score, the better cognitive function it is. And the other surveys are the opposite direction. Brain injury visual symptom survey, there has been already literatures available studying post-COVID syndrome particularly, and you can see the detail of the questions about visual disturbances, and our cohort, all Meister study cohorts show the greater symptom burden in clinical cohort. And then fatigue is the same thing. It is just so clear the symptom burden is so great. Now, I would like to talk about the most commonly used clinical tools when people go to like a physician, like primary care provider, talking about these symptoms. So, I used to be an emergency nurse too. When people come up with like a disorientation, some things that we will immediately use is like mini mental state examination or general cognitive assessment. Can you draw the clock? What's the date today? And could you repeat the word? And can you recall that word after a few minutes? Well, mini mental, same thing. What season are we? And then, or, can you just, what object is called, or what object is called? Those are the questionnaire that we ask. And the battery testing that is remote cognitive testing that we apply a day before participant arrive and then a week afterwards. Because with the infection-associated chronic illnesses, there's a lot of PEM, post-exertion malaise. People are different when they come to study visit, and 3 days afterwards, their symptom flare-up starts, and 5 days it can be worse, and 7 days it can still linger. So we try to understand this battery score a day before and then a week after study visit. I would like to share this right eye test here. I don't know if it will play here. Okay, it's not playing. I'm very disappointed. But eye tracking testing, I can verbally explain. You see this solar system here, and then what we ask to participant is, I have them understand the instructions shown on the video game like testing. And then they're gonna see the solar system, and then one image will pop up. If you see alien, press 1. If you see astronaut image, press 2. If you see Earth image, press 3. And the images keep rotating. I don't say anything because I don't want to intervene that testing, but participant is supposed to process the visual stimulus and be able to process and react correctly by answering the correct answer. And also sometimes we'll give a different cognitive testing applying this eye tracking test. And we also check a very simple ocular motor function by having these people look at the dot and do the circling motion or saccade, look back and forth. It's just a basic ocular motor function we try to look at further. And I will share with you guys the raw data that is quite striking later on. And the EEG is we are doing the commercially available EEG called WAVI, and device looks like this. And in the EEG testing, we also apply cognitive processing testing. It's called audible paradigm, which is also commonly known P300. A person will hear 2 different sounds, and one is high pitch, one is just common tone rare sound. And high pitch sound we play randomly. Every time a person sees a different sound, hears a different sound, supposed to tap the space bar. So it requires that cognitive processing, but we'll also be able to measure their simple reaction time after the stimulus. So I mentioned about symptom burden that is already great, like explained, it's just a significantly different. Let's go over some interesting results here. A general practitioner cognition exam. It's a question that I mentioned earlier. And as you can see, healthy control, long COVID, and chronic Lyme, they all show the similar pattern. Half of them are perfect score, 9, no impairment. And then half of them will be like 5 to 8. 5 to 8 is considered mild or moderate or more information needed. That's how they interpret it. Mini mental, a lot of them are just fine because mini mental state exam, the score is all normal, no impairment if one gets score higher than 24. 19 to 23, you have to have several questions wrong, but in our study cohort, only 3 in long COVID has the MMSE requiring more information. Now, the reaction time, simple reaction time, when we hear this has said in 2 different sound tones, we are seeing that the slowest group, the interquartile, slowest group of control group, compared to the control group, long COVID and chronic Lyme, 3% and 53, more than half of the percentage people is slower than the slowest group in our healthy control. Same with the eye tracking testing. 44.4% in long COVID and 52% long Lyme is slower than the slowest group in healthy control. And same with the brain check battery testing. What I try to say here is battery and eye tracking and EEG testing, those are very different test modality. However, we're observing similar pattern when we measure the similar reaction time variables. So I think we wonder if there are any potential ways to measure these subtle changes that happens after infectious disease. I would like to mention about case study here. 54-year-old female who got Lyme disease August 23 came to came to MIT and get tested. 9 months later, her neurocognitive changes was so significant that she was too sick to be able to even come to the 1-year visit that I invited. Her most complaint is the neurocognition. She tried everything that she could get back to work. Hyperthermia, tried all kinds of methods, but nothing worked. When we compare her visit 1 baseline when she had acute Lyme, her reaction time, and the yellow highlight one and the below visit 2 shows the delayed reaction time that we were able to capture from our study visit. Another acute Lyme, 26-old, the young female, highly functioning student, 6 months later after acute, she reported physically about the same. And, we had a second visit. And then 1 year later, very recently, she feels overall better. She says she feels better. However, she has told me that she had recent diagnosis adenomyosis and PCOS and just gynecological procedure. I wonder if this is changes that happens to after acute phase. It's too early to make any conclusion, but these are the data that we think is absolutely precious and really start to analyze further. I want to share this smooth pursuit eye tracking testing because first one is baseline, middle one is a 6 month. At 1 year, even though she says she feels better, when we run this testing, there are some changes being noted. So we wonder, even the patient reported outcomes say, I feel better, I feel worse, we could collect the data, but how can you go beyond the patient reported outcome when there may be some functional structure damages might still be happening, and how do we capture that? So we're constantly learning from our study visit. Control and chronic Lyme. The video is not playing, unfortunately, but this is the eye tracking data that I was talking about. I know how strikingly that is different. This person is particularly very, very sick that I remember up until now. I want to show one last thing here. The table on the right side, you see all multiple commensals, GI commensals detected from our carriers testing, the pathogen DNA testing. So when we run it, we see this list of commensals, and I put it to my participants and say, as a research nurse, I cannot make any medical diagnosis interpretation, but I can give it to you. So you please go ahead, talk to your doctor about this. She found the infectious disease doctor and then presented this list. And she shared tears with me and told me that she thought she was gonna find some answer from her devastating illnesses, but infectious doctors say, if you truly have all of this listed, you'll be very sick. So just wanted to share that. And again, this is her data. Recently with the proteomic data from Olink, the GFAP is very high. She's marking there. I deleted the patient ID. And that correlates to the reaction time that got slowed down in our eye tracking testing. The NFL is also, she's marking above there, and she is one of the slowest group. So we wonder if there's any potential way that we can integrate this proteomic data and correlate to the objective neurotest measurement. So just wanted to share that. I put this slide just in case the video didn't play, but I'll skip it. Here's my summary. Brain fog is more than a symptom. It's much more complex here. Of course, infection-associated chronic illnesses are always complex. We have to look at the systemic, the multi-angle approach. And I presented this in the very beginning, what we measure here. And this I also want to share, and I will disappear shortly. Blood flow, vascular dynamics. We wonder all this Olink data that we're seeing, and then the neurocount data we are getting, is there any correlation that the vascular structure be, relate to their poor cognitive function too? And if so, can it be captured through the capillaroscopy? So we're also actually doing the capillaroscopy, and you see the ear picture there. We're doing the Lumia earpiece that measures a proxy cerebral blood flow. So is there any correlation that we can measure beyond that, just the classic neurocognitive testing that we do? This left picture is a recovered normal capillaries that you will see. The right one, as you can see, the 65.4% is a high percentage of enlarged capillaries. Again, with that said, I would like to thank you everyone, and then I'm just very grateful for this opportunity, and I'm thankful for my team and Dr. Tao. Thank you so much.
Thank you, Yuri. Now I think we have some time for questions for the speakers from this afternoon. So, Nikki, you said somebody was on chat. Was that Lavinia?
Yeah, this question.
Okay.
So for Tomas, Lavinia posed a question in the chat. Did you find agglutinating proteins in the gum or only in brain where insoluble proteins can't be cleared?
Bob, you have a microphone there. I believe.
We also have the other mic. Not sure
Oh,
where the other mic
yeah. Okay, so thank you for the question, but we didn't really check in the brain. So sorry. Okay, so yes, we didn't check in the brain, so I can't answer really to this question. Sorry.
So in case it wasn't audible for everyone in the virtual platform, they didn't check. So we can talk more about that next time as a team when we're all together on Zoom. Brent.
I have a quick question for Yuri. Really great presentation, Yuri. Right here. My question is, I know you're early in your cohort discovery and collecting patients. You have a fairly large age range, and I'm curious to know if you're finding differences by decade.
Hello. Hello? Okay. So your question is if, if you consider all the age dynamics, right? Absolutely. Because their age plays a factor too, of course, obviously. But it's been interesting for us to also see, despite the age, people we're also, when you think about the school age students, college students who got very sick with after acute Lyme disease, we are seeing striking delay. And I first thought that age will be the factor that will be very confounding. And how do we think about this? And we kind of have to really dissect this age different group and then, their comorbidities. We have to also consider severity of illnesses and illness durations. Everything matters. And I do, beyond age, I think those illness duration and severity really plays a key role there, because that's why we're seeing this impairment so significantly in early 20s and 30s. So while that is always in our mind, I personally think that there's something beyond that age cannot really, interfere this result data so much.
One more quick one. That the data with the capillaries is fascinating. You threw that in at the very end. I can't wait to see how that turns out. Are there similar capillary changes in both long COVID and in Lyme disease?
It's unofficial, but this I want to share my personal, because I've been the nurse who's doing the capillary use for so many people. Part of my insight is that, again, this is when the illness duration, I think, plays a role too, because COVID with the pandemic, it's been, let's say, 5 years, let's say so. But our chronic Lyme cohort, I divide a group in our study, 1 to 3 years to 4 to 10 years and beyond 10 years. What are remarkable, noticeable structural changes that I see in people who suffer from this for more than 10 years. So that's something that I absolutely have in mind. I don't particularly see long COVID chronic Lyme. Are they different? Not really, but I keep thinking about how many years they've been suffering from this and the complexity of their comorbidities too.
So following up with the discussion on the capillaries, there's a fair amount of research that's been done by people studying chronic Lyme using SPECT imaging, showing acute vasculitis in the white matter. This is like work that Francine Hamburg and her team has done out in Los Angeles, and then Bob Mosiani has also shown this, that there's very pronounced white matter vasculitis you can pick up by SPECT scanning in any of these neurologically impaired Lyme patients. I don't know anything about long COVID, but for Lyme disease, this is actually very well characterized.
It looks like a lot of the symptoms that you check for in your testing are also applicable to chronic fatigue. Do you ever deal with chronic fatigue patients?
Yes, and I have a lot of thought into that. Chronic Lyme cohort patient, a lot of them also end up diagnosed with the ME/CFS, chronic fatigue syndrome. I do think that's a part of the illness journey that they ended up having. So while I know that these are part of the consequences after chronic Lyme, some studies kind of exclude that in the ME/CFS criteria to make it very clear it has to be just related only Lyme. But I just think those comorbidities that gets involved with this type of chronic infectious disease is so complex that I do think we cannot just exclude that cohort particularly. And we're going to later on really like subgroup of people who has a ME/CFS criteria falling into the criteria and who does not, and to also see the differences from their biomarkers and the other data that we do. Yeah.
You maybe sent the lines to your physicians. Is that correct?
Yes, so that's one thing that we try to make a difference. When have participants spend that much time to travel all the way and really provide us the precious data, I think as a return for our study team is to give them as much information as we can. However, I'm a nurse and I know that the danger zone to when we give out all the information, I just want to make sure that these are the clear certified, like cleared reports that we can generate from the companies. Those are the risks that really would be very costly from out of pocket from patients, but we were trying to generate those reports and give back to participants. But again, we do not make any interpretation for that. We can, unfortunately, and let us just publish with the exciting data that can help the community. But to us, we're just giving out, and then they have to discuss that with their physician.
Hi, for Nikki Bell. Comment, all the skin diseases that I've studied, and I've studied a lot, are biofilm diseases. Was any effort made to approach this for your husband with a biofilm buster along with an antibiotic? Including rifampin?
For everyone on Zoom, Nicole Bell said yes, that this was tried. Any other questions?
I have a question for her name, Yuri. When you do the capillary studies and you notice that there's blockage in the capillaries, are y'all studying any particular pathogens associated with that, like the Lyme and co-infections, or are you seeing it associated with certain ones over others, or is it just sort of generally?
I'll take that one. So, I mean, our cohorts are broken up into comparing either Lyme or COVID. In our Lyme cohorts, we do have people with co-infections, but they have to have had Lyme. So Lyme has to be one of their infections. They can have others. We haven't yet done that subgrouping of, if you only had Lyme or if you had Lyme and Babesia, would that matter for your capillaroscopy? I wanted to mention one of the most important things, and we didn't have time to show this, but we do that capillaroscopy on fingers and toes before and after the nasoline test. And that's where people who don't have symptoms on the nasoline test, we're not seeing any differences in the capillaries, and we wouldn't have expected them to be very dynamic. But the people who have symptoms on nasoline tests show extremely dynamic changes in their capillaroscopy before and after, and especially in their feet. And so that's just something that just adding in that perturbation can really, bring out these extremely profound and striking results in a capillaroscopy. And you can see vascular leakage and hemorrhaging. So, and this is all, painless and non-invasive. And I think that that's somewhere where on kids or people who, you can't necessarily have them consent to different medical procedures. This is something that we really should be taking more advantage of.
We can take a short break now, and then we'll have an opportunity to have more open discussion with the panel. So we'll come back here in, 5 or 6 minutes.
Okay. Okay.
Thank you.
I'm on the board. We have some Parkinson's, we have some molecules vaccine. That's very awesome. Yeah. Listen to me.
Let's get, we're gonna do another picture.
But I'm good.
I have to be on the right hand side.
Positive. Okay.
So, yeah.
Okay, cool. Thank you so much for coming over.
Yeah.
I'm glad I could do that. Yeah, I don't even.
Other So, you know, there was like. We're very interested in understanding or is this a multiple that reaches a point of discretization.
Hi, everyone. I think we'll get started with the rest of our sessions and speakers. If everyone can take their seats, thank you so much.
Just wait for me. Settling down. Wonderful. So our next speaker is going to be Brent Harris from Georgetown University. Welcome, Brent.
Thanks. You guys can just call me Brent. I want to thank Brian and Nikki very much for inviting me to come talk. And I also want to thank Nikki, Nicole, and Susan, who I really admire the strength and tenacity and ingenuity of what you three women have done in your adversity. Really impressed. Okay. Nobody else did disclosures. You guys all pure? I guess so. I am too. But I do like to, disclose some of the boards that I'm on and the funding that I have as well. Some of it's relevant. I mean, it's all relevant. And this is my most important slide. I like to dedicate this lecture to the patients, families, and caregivers who battle neuroimmune disorders. So I'm going to talk about brain disorders and neuroinflammation, brain banking, and the establishment of the Pond Brain Bank, which I'm really proud of, and then a case discussion. And we won't have time for questions. We'll do questions at the end. I forgot we're going to do that later. What do neuropathologists do? All right, how many neuropathologists in the audience? Raise your hand. Don't be shy. All right, well, you all picked the wrong profession, okay? And that goes for anybody out there, although there might be some neuropathologists out in the cloud. It is the best job and most interesting thing in the world. And I'm really happy I've done it for my whole career. We study and diagnose and teach about ailments in the brain. We don't treat them, but our diagnoses are really key to treatments. We study neurologic diseases, trauma, developmental abnormalities. A bunch of us are brain bankers too. We're often teachers, consultants, and researchers. So I'm going to keep the talk at a fairly simple level and not do a lot of hard science, although I have been incredibly impressed by the very cool science that's been presented today. But let's talk about generally what's inflammation of the brain or neuroinflammation. Well, it is a bunch of different things, but it probably, I'm not going to say it starts, but it's certainly influenced by circulating immune cells, lymphocytes, macrophages, and all the other immune cells coming from the blood. It's influenced by circulating molecules from the blood, antibodies, cytokines, chemokines, other proteins, exosomes. And then there are intrinsic cells within the brain that are very important in neuroinflammation, and they also secrete molecules. So every cell in the brain can be involved in neuroinflammation, including vessels and perivascular cells. Here's a picture. It's kind of bright in here, so fluorescent pictures don't show up great. But this is a corticospinal neuron from a mouse model that I studied many, many years ago during my postdoc. You can see the beautiful axon coming out here, the apical spine, and this one's in focus, and it's kind of sitting in a layer with others. And I can't remember who said it, but neurons really got most of the attention for a long, long time. But I'm a glial biologist. And all of the other cells around, all these little blue dots are just nuclei of other cells around, are equally important to the neighborhood that these neurons are working in. So some pictures of inflammation and some of the players. So normally we don't have lymphocytes in our brain, although they can circulate around the CSF. If you have too many of them or you have the wrong types, that's where problems come about. So you can see this blood vessel here in the middle. Oh, no, you can't see my pointer. Okay, the blood vessel in the middle with the red blood cells, and then all of those little dark blue spots around it. And so they accumulate and they come from the blood vessel. But even percolating within the brain tissue, you'll see some lymphocytes too. Reactive astrocytes. These are what reactive astrocytes look like. These are cells that become reactive as a response to either trauma or some other cytokines, infection, stroke, a whole different host of subjects. And the same is true for microglia. They are your resident immune cells, and they actually have the ability to move around a little bit regionally to pick up bacteria or eat up dead things. And then macrophages can come in either from the periphery, or microglia have the ability to assume macrophage functions. So what conditions elicit neuroinflammation? Not vitamins, but that's an acronym that I use with my medical students. So about anything you can think of can induce neuroinflammation. And here's the acronym. We take a little liberty with psychiatric. CNS infection syndromes, these are range, you've heard about it today, from trivial to fatal pathogens. And the cardinal clinical manifestations are fever and headache, altered mental status, and focal neurologic signs. These are all the things we've been talking about today. Here's a whole list of various different things that can cause problems in the brain and other areas. We also have entities of post-infectious encephalomyelitis, where there's an autoimmune, sometimes demyelinating, but not always. And these are from mimicry of antibodies being made against antigens of the organisms or viruses. And oftentimes with encephalomyelitis, the CSF is sometimes normal or nonspecific, and MRI can sometimes show things, but not always. Here's what encephalitis looks like in a fairly acute stage, where you see collections of lymphocytes and other immune cells collecting in the brain parenchyma and some blood vessels that don't look very happy. Here are those perivascular cuffs that I showed you from before. And then here are microglia that are collecting in the middle there, that are trying to eat up some viral infected cells. Sometimes you can see the viruses directly. These large nuclei in those cells are viral particles, and we can use immunochemistry to identify them. And infectious disease can be devastating and fatal. This is an example of herpes encephalitis, where you get this hemorrhagic, terrible infection, often in both of the medial temporal lobes. Here's an example of an autoimmune encephalomyelitis that is knocking out a bunch of the myelin in areas. So non-infectious or autoimmune, you can have tumor-associated or you can have post-infection associated. You're not going to be able to read this. Well, maybe you can. But these are a lot of the antibodies against, they're often against neurotransmitters. And they can be elicited by tumors or infections. Because the antibodies are against neurotransmitters, you can understand why they might have a lot of neuropsychiatric type manifestations. Here's the laundry list of post-infectious encephalomyelitis organisms, not exhaustive. And we have had the privilege at Georgetown of studying a variety of different things in the brain, and having funding from nicely from the last year from AlzPI and PCOM and the Gosnell Foundation to start doing sterile autopsies. And so we're developing ways of collecting the tissue in a very sterile way that neuropathologists generally have not done in the past. We have another grant from PolyBio Foundation to establish a rapid autopsy and obtain multiple different tissues to look for persistent SARS-CoV-2 and long COVID syndrome. Both pretty early starting grants, and I would love ideas from everybody about how to perfect them. So brain banking is a really important aspect of what we do in academic neuropathology. Anybody in this room could donate their brain when the time comes. I wouldn't do it now, but you can do it when the time comes. It's an easy thing to do, and it's getting easier And if you have a particular disease, then often there are brain banks associated with it. If you have no disease, which I hope will be true for everybody in the room, you can still donate your brain. We need lots of people to donate who have no neurologic disease. You're not a control. None of us are a control. We all have different genetics and life experiences, but you can be compared, your tissue can be compared against other diseases, and that's an incredibly important thing. To do that, actually, the NIH has a program called the NeuroBiobank. If you just go on to the NIH's website or NeuroBiobank and NIH, you'll find it immediately. And you can kind of pre-register to be a brain bank donor, and they'll help orient you to what you need to do. So when we brain bank, we are doing a good neuropathological evaluation so that we know what the diagnoses are. We know what the clinical diagnoses are, but we want to know what the neuropathologic diagnoses are. And then we can store the tissues either frozen in paraffin, in slides. They can be stored extensively in lots of places, or they can, be at each institution. And I've done it kind of both ways with some of our consortiums. The frozen tissues, we have to take a little more care with, but we can do all kinds of omics. And I'm going to now break to talk a little bit about the PANS, PANDAS, and other neuroimmune disorders pond brain bank that we established just a few years ago with the support of both Georgetown and philanthropic support from the Alex Manful Research Foundation. So our mission is the mission is that the Pond Brain Bank was founded to aid researchers, medical students, clinicians, our patients, and their families to advance understanding of PANS, PANDAS, and other related neuroimmune disorders. Our goal is to assist research efforts to ensure that no person will lose their life to PANS and PANDAS or lose the years that it can take to successfully treat these neuroimmune disorders. So currently, we have 9 cases in the bank. These are most of them all really incredibly sad cases of teenagers or young adults who have taken their life. So we're working closely with medical examiners around the country. We get referrals from families who have lost someone, and then we work with the medical examiner and try to obtain the tissues for the bank. I'm going to describe a case now. So this was a previously healthy 19-year-old female who had mononucleosis and strep infection, new allergies, new plaque psoriasis. She developed new neuropsychiatric conditions early when she was in college, which worsened to the next several years, and had OCD, depression, insomnia, eating disorders, and alcohol use. After graduating from an Ivy League school, she worked in several prestigious high-stress jobs on the East Coast, and she was diagnosed with PANDAS at age 26. Several months later, she seemed to have an acute episode of fever, aches, and went to the urgent care. She seemed to be doing better and communicating with family, but then tragically took her life. So we were asked to take over the custodianship of these tissues. The case was handled by the local medical examiner's office and sent to NIMH. And in 2020, the family requested the biobank slides, brain and blocks be sent to Georgetown for further review and research and teaching and distribution, eventually to the research community. So when we looked at the reports and looked at the tissues, we found a normal weight brain, normal meninges, vasculature was normal, maybe a little bit of early patchy atherosclerosis. The vessels are incredibly important in all brains, and even young people can have early atherosclerosis. So watching what you eat, keeping your cholesterol down, and your hypertension controlled is incredibly important for brain health. And really, they did not see any brain abnormalities when they did the brain cutting. The neuropathologic microscopic evaluation showed H&E findings that were largely normal, with the exception of findings in the basal ganglia and thalamus. occasional vessels with mild, I would say, to moderate perivascular lymphocytic cuffs, scattered metabolic glia, and mild to moderate reactive astrocytosis. No demyelination, no abnormal tau, no viropathic changes, and no microglial nodules. So here is what the tissue looked like in the basal ganglia. You can see these thin cuffs of lymphocytes at this low power around these expanded vessels. We characterize the immune cells to some degree, although I would say that there's still more work that can be done on these. And we found that there were mostly T cells around the vessels. CD3 and CD4 are both T cell markers. Not as much CD68 and very few B cells. We did a little bit of immunohistochemistry, and you can recognize the CD3 and CD4 and CD25. CD25 is a marker of IL-2 and often seen on T regulatory cells. This is probably the second most important slide in the section, though. So this is her basal ganglia, and you see the two vessels in the middle there. And around the vessels, you see an accumulation of brown material. And that material is called glial fibrillary acidic protein. Normally, astrocytes, one of their normal functions is to put down little foot processes along blood vessels, and they play a role in the blood-brain barrier. And so when you have inflammation and astrocytosis, that can affect the blood-brain barrier. So we're looking at kind of the Empire State view down at these vessels, and you really should not have this degree of inflammation within this region. And this is what's known clinically when imaging is done with kids and young people with PANS-PANDAS. So we think that this gliosis maybe is playing a role in the blood-brain barrier and in the pathophysiologic process in this disease. We published this case report. We think it's the first neuropathologic evaluation published for PANS-PANDAS. Sadly, we're seeing some of the same change in some of the other cases that we have in our bank. So the future markers that should be looked at include other T regulatory markers, more research on the blood-brain barrier proteins, and then spatial transcriptomics, I think, is absolutely necessary to try to understand the genes that are playing a role. And so we can learn a lot even from one case about this disease, and then go back to animal models to try to parse out treatments and other questions. So this is my last slide. My contact information, our coordinator's information, and feel free to contact me. I'm looking for researchers who want to use this tissue. Brain bankers always need to collect the tissues, and we have biofluids on some of them as well, but we want to get them in the hands of research teams. Thank you.
Thank you, Brent. Our next speaker is going to be Lynn Bimler from Baylor College of Medicine. Welcome, Lynn.
Thank you. And thank you, Nikki, and everyone for the opportunity to present our work. Hopefully everyone's not too tired from a day of awesome data and too much excitement. So I'm going to present my research project on Candida albicans gut colonization derived chronic polymicrobial cerebritis causing neurodegenerative disease in mice. As we all know, over the past few decades, especially, there's been increasing focus on the idea that various pathogens, including HSV1, C. pneumoniae, spirochetes, H. pylori, periodontal pathogens, and our favorite in our lab, the yeast Candida albicans, as well as many other things that we've been discussing today, have all been linked with Alzheimer's disease. Our lab produced a acute animal model published in 2019 where we gave an IV infection of Candida albicans and found that our mice had transient Candida cerebritis as well as slight memory deficits. And so our goal moving forward was to create a chronic model through gut colonization. And so what we did was give an oral gavage of Candida albicans, hoping to colonize the GI tract, and that this would result in leakage of Candida albicans into the bloodstream, which had previously been reported in both humans and mice. And that because of our study of systemic Candida albicans going into the brain, we thought that this would lead to brain infiltration and hopefully eventually cerebral mycosis and eventual dementia. To save us some time, I'm just going to let you know we can colonize the guts of mice. And then I want to dig deeper into what we're seeing in the brain of these mice. One of the most startling things we found was that when we look at our yeast inoculum or at the brains of IV infected mice, the yeast looks morphologically like a yeast. So macroscopically, we have a nice round fluffy colony, and microscopically, we have yeast cells and maybe a few filaments or pseudohyphal cells. But when we looked at the colonies from these oral gavage mice, at first I was like, our experiment has gone horribly awry because these colonies did not look like a classic Candida albicans colony. They were white but patchy with yellow and morphologically irregular. But I put them under the microscope, and what I found was that these colonies were actually polymicrobial biofilm consisting of a sea of bacteria combined with larger yeast cells, some of which you can see are actually budding yeast. And we also have filaments and pseudohyphal cells. And so this was really striking. We even see some cells, and this is maybe not the best example, but we see some where it looks like a fungal cell with smaller bacterial cells attached to it. And so we really had to revise our thinking here and consider that perhaps after we do the gut colonization, what's actually linking into the blood vessels and eventually the brain is a polymicrobial unit consisting of Candida albicans and bacteria. And this has actually been noted in the brains of Alzheimer's patients before. So we felt like this was along the right track. So we looked at the brains of these mice more intently, and we found that they had the same FIG structure or fungal-induced glial granulomas that we had previously seen in our acute mouse model. And so here we have a serial section with calcifluor white staining indicating the presence of fungi. And then in a serial section, we see a foci consisting of A beta deposition, shown in red, as well as activated microglia clustering around that foci, and that's shown with IBA1 positivity in green. And then you can just see the accumulations of cells in that region with DAPI. And so this we called a FIG, and they're present 2 to 6 months post-infection. And we haven't checked earlier than that or later than that in this model, but presumably if it's happening for 6 months post-infection, it's probably a continual process. So our FIGs are present in the brains, as I said, between 2 and 6 months, but there's not a large number of FIGs. It's pretty low level. So here, looking through the entire forebrain, we see an average of about 5 FIGs per mouse brain. But you don't see any of these in the naive mice. They're only seen in the infected mice, and they're seen at all time points. We also see them consistently in the same regions of the brain out to 6 months. So they're seen in the cortex and corpus callosum, the hippocampus, and the hypothalamus at pretty consistent percentages. And by 6 months, these mice have a pretty striking loss of brain volume in specific regions. And you can see that has a lot of overlap with where we are finding these FIGs. So we didn't actually look in the pons medulla for FIGs because we only looked in the forebrain, but we have decreased volume in the pons medulla, as well as the thalamus, hypothalamus, and superior colliculus. And the hypothalamus was one of the regions that we saw FIGs. And the cortex also has decreased brain volume, where we also saw FIGs. And all of these regions are also linked to AD, and specifically early AD in humans in the case of the corpus, the hypothalamus and the cortex. And we also see a trend towards increasing ventricle volume, which is also associated with Alzheimer's disease and neurodegeneration, because as you lose brain volume, the ventricles get larger. Looking at what might be causing this loss of brain volume, we think a loss of neural synapses. Here we're showing a decrease in PSD95 staining, which is also confirmed by Western blot in our infected mice. So we have less red, which is showing PSD95 here. And we also have a decrease in dendritic spines, which are the primary site for synapses. And so all those little spikes that are coming off of the main dendrite are sites for high levels of synapses, and we have a significant decrease in those mice. We also see increased numbers of microglia cells throughout the CNS. So this is in the retina. We have an increased number of microglia. And then if we look to the cortex, prefrontal cortex, we also see an increased number of microglia, and they have a decreased cell process length, which tells us that they are more activated. So what we're showing here predominantly is that there are big changes in what the brain structure and pathology looks like. And specifically, we think that we're both losing neurons and neural synapses. I haven't shown the data showing they're losing neurons just for time, but we've done RNA sequencing and we can show that specific subsets of neurons are actually decreased in number in the infected mice. And they also have overall highly altered transcriptional profile in several different neuronal subtypes. And so how is all of this affecting the brain? Well, we looked at the long-term potentiation of the hippocampus, and we found that it's completely abrogated in infected mice. So these mice were 4 months post a single infection, and remember, these are wild-type mice. And we found that the mice that were infected, their LTP never really got above baseline for the test. So the measurements in the far right graph are actually the measurement that matters. The other test is showing that the test, or the other graph is showing that the test worked. But because you basically prepare the neuron by performing the LTP assay for over an hour. And then once the neuron has kind of been primed, those final reads in the last 10 or so minutes are the ones that actually matter for showing if the neuron is functioning or not. And here you can see it's basically never going above baseline for the red neurons. And so finally, we wanted to see if these changes in the brain pathology and the retinal pathology and the brain function also had an effect on the mouse's behavior. And so we did a novelty-induced grooming test where normally the mice would groom in a novel environment as a result of anxiety, but our mice actually were extra anxious and would try to escape the chamber by jumping up and down. So we actually counted the number of escape attempts as an indicator of a kind of more severe anxiety. And at 4 months, we had a significant difference. By 6 months, it's borderline significant, but it does seem like the control group goes up, which is a trend we've seen a lot in some of our tests. So we think that perhaps some of these are a function of natural aging, and we're just having faster phenotypes in the oral gavage because of the results of infection, but maybe the naive also start having a little bit of this anxiety just as they naturally age. We also, so we did 8 tests, but I'm showing you 2 of the significant ones. We have a couple others that were different, but we also found that they had memory deficits in the conditional fear test. This is a test where you put them in another novel environment, allow them to explore, and then after a few minutes, you electrify the floor, and then you rest the mice for 24 hours and place them back into the chamber the next day to see if they recall being shocked. And a fear response when being placed back in the chamber would be to freeze. And at 4 months, you can see we have significant lack of freezing in the infected mice. At 6 months, that trend goes away a little bit. And finally, just looking, is this similar to humans? Obviously, a lot of the pathology is reminiscent of human samples or human patients, and as well as some of the behavioral changes. But this polymicrobial growth that has been reported, at least scarcely in the literature, a little bit, we wanted to see if we actually culture human brains similarly to how we culture mice brains, do we get a similar result? And a high number of our colonies that grew out of human brains are polymicrobial, consisting of acetobacteria as well as yeast, some of which are budding or pseudohyphal. And again, some of them even have smaller bacteria cells kind of accumulated on these fungal elements. And we've also looked at this not just through microscopy, but through scanning electron microscopy. And here we can see that there seems to be this accumulation of what looks kind of like fungal filaments, the spaghetti in this image. And then we have smaller circles, which are bacteria or fungi. And you can tell the difference because the fungi are about 10 times larger than the bacteria, as would be expected. We've also gone forward and confirmed the presence of fungi and bacteria in many of these colonies through a combination of ITS2 and whole genome sequencing. So finally, we feel we have developed a model where we can oral gavage with Candida albicans in a wild type animal, and this leads to gut colonization and through leakage into the bloodstream, brain infiltration and polymicrobial cerebritis and eventual dementia. So we think we've developed persistent polymicrobial cerebritis causing devastating neurophysiologic impairment that manifests as increased anxiety, memory loss, and retinopathy. The human AD, but also non-AD brains contain similar Candida albicans and other fungi-based polymicrobial affiliations. And so we propose that Candida albicans could be a model organism for a potential microbial cause of degenerative brain diseases and used in wild-type mice. And we think this is very similar to Alzheimer's disease. So I just want to thank my lab and our funding sources, and look forward to questions at the end. Thank you.
Thank you, Lynn. Our last talk for this afternoon is going to be from Dana Cairns from Tufts University.
Thank you, Nikki and Brian, for the invitation. It's been a very lovely day so far and very informative. Let's see, just do this in case I get mixed up. Okay. Yeah, so thank you again. And I just want to mention I'm at the lab of David Kaplan, and I work very closely also with Ruth Itzaki at Oxford, who's been just an incredible mentor. I'm very grateful for her knowledge. So I won't get at this is old news for everyone here. We've already gone through a lot of this. I keep this on here just to highlight some of the markers I'll be looking at in our system here, which have already come up. Gliosis, so GFAP is one we'll be looking at. And beta amyloid, fibrils, thioflavin T, as well as phosphorylated tau. And again, this is why we're all here. What causes AD? What causes these types of neurodegenerative disorders? And from my perspective, I'm really focused on the role of herpes viruses, in particular HSV1. And this has come up quite a bit in terms of causality. There's quite a number of population studies that support this idea, as well as follow-up studies in my lab. And again, Ruth has been a real pioneer in this space. As a self-described heretic, she's met quite a bit of pushback over the years. So in our lab, again, my lab is biomedical engineering, so very basic science. We have a lot of tools at our disposal. And my goal was to really first kind of investigate, is there a potential direct causal role of HSV1 in AD pathogenesis? And again, we really took advantage of some of the tools we have in our lab. One of the things in our toolkit we have are HINSCs, or human-induced neural stem cells, and this was a technology developed there, which has since been out-licensed if people want to buy it. That's just a shameless plug. But these cells are different from iPSCs in the sense that they're a little bit more downstream. They're potentially, they're held as neural stem cells indefinitely. There's no pluripotent intermediate, and they're really good for a lot of these high throughput studies that we do in our lab. So I really just wanted to understand initially, can HSV1 infect HINSCs? Is there any correlation to AD? And ultimately, we culture these in 3D models in our lab. So HINSCs were, in fact, quite infectable by HSV1. One thing I want to point out relative to some other systems is the fact that the infection rate, the MOI I have to use in these, or the multiplicity of infection, is quite low and physiologically relevant. Typically, for these experiments, it would be 1 viral particle per 10,000 cells, which is, it results in a productive infection, which is what you would see in a human condition. Again, GFAP, a marker that's come up, this is just showing that in response to HSV infection, you do see robust reactive glia expression, as well as kind of the classic neuroinflammatory markers, IL-1 beta being one of the major players here. And one thing that we noticed kind of early on was that you visualize these really large plaque-like structures, which kind of look like amoebas, and those are the result of these multicellular formations or syncytia that form in response to virus. And the more differentiated the cell, the smaller these plaques form, which in our hands we kind of use to equate to immaturity of neurons. So the more immature the neuron, the more profound the AD-like response. And if you stain these things for, classic amyloid beta, thioflavin T, and phospho-tau, you see that those plaques specifically express really robust expression of these markers. And so for most of the experiments done to date, we've used a HSV strain known as McIntyre, which is quite common. There are also a number of other strains that have different rates of neurovirulence. And one thing that I wanted to just kind of highlight here is the difference in syncytia formation, which I think is relevant in terms of thinking about how this virus can wreak havoc in different ways. And particularly, you can see that this strain here, which is also clinically relevant, has much higher phospho-tau expression. And so again, in our lab, we make use of these 3D brain-like tissues. We use these scaffolds, which are quite literally sponges made out of silk, which are seeded with NSCs and infiltrated with collagen to allow for neuron infiltration. And we call them donuts in our lab. And if you infect these donuts with HSV1, again, at similarly low levels, you see what we saw in 2D, which is robust amyloid beta expression. And we also have the capacity to visually look at neuronal firing within the window of these donuts, which is really nice and gives us a nice kind of high throughput way to not only test to see if these viruses are doing, having negative effects, but in terms of understanding any kind of protective or therapeutic effect, this gave us a really nice platform to do that. And if you look at some of the differentially expressed genes, this is just a commercially available panel that's from QIAGEN. We found that quite a few genes associated with AD were differentially upregulated, 40 specifically, and some were downregulated, which again kind of aligned with this idea that HSV1 can on its own really induce this robust effect that's similar to AD. We also want to understand, are there any potential indirect causal roles of herpes viruses in AD pathogenesis? So by that I mean, are there other pathogens or other mediators that could potentially induce similar phenotypes? We know that the correlation between HSV1 is so profound. Maybe there are other pathogens that can have similar effects. And one that came up in our screen here is varicella zoster virus. As probably a lot of people here are aware, the shingles shingles is caused by VZV. And a lot of studies have come out recently showing that vaccination against VZV actually results in a significantly reduced risk of dementia, and that's across different types of vaccines with Zostavax, with Shingrix, really alluding to this idea that if you can prevent VZV infection, you can also prevent potentially dementia and Alzheimer's, which is pretty exciting from our perspective, and this was in collaboration with Ruth as well. So similarly, we wanted to understand if VZV can infect HANSCs, and it actually can. which we were excited about because it's been known to be a little bit trickier of a virus to work with as it's not productive in the same way that HSV-1 is. We also found that similar to HSV, VZV also induced GFAP and inflammation, which is not surprising either. So one thing that we found out, which we were not sure would be the case, was that unlike HSV-1, VZV on its own did not actually induce this robust beta amyloid phenotype. However, the combination of both viruses did seem to have an additive effect, resulting in plaques that were much larger than HSV alone. And again, there was no p-tau expression in response to VZV in our system. So this is kind of where we shifted gears a little bit. We wanted to understand the potential role of secondary VZV infection. By that, I mean, if you were to have a primary HSV infection and cause that infection to become latent by use of antivirals, in this case, we use valacyclovir or Valtrex, to silence the initial HSV infection, we saw that, so just to kind of walk you through this panel a little bit here, I think it explains it a little bit better. Again, so you have the HSV infection followed by antiviral Valtrex treatment and a secondary VZV infection. You see that this secondary infection with VZV actually reactivates HSV. And similarly, you see this robust induction of A beta, suggesting that this secondary VZV infection is actually what could potentially be causing the dementia and maybe not necessarily a primary infection itself. So kind of moving forward from there, we wanted to look at other potential sources of inflammation, since we know that VZV on its own can actually induce robust gliosis and inflammation. We want to understand if other secondary infections similar to VZV, other herpes viruses, other types of pathogens, bacterial derivatives. And we were also really interested in the role of injury in this model. So for these subsequent experiments, we used similar reactivation studies. And HHV6B is a beta herpes virus, so a different family of herpes viruses. And in these studies, this is unpublished data, we also saw gliosis and inflammation, but similar to VZV, we did not actually see specific AD phenotypes in the form of beta amyloid plaques. We did see kind of an additive effect of co-infection, but other than that, it wasn't particularly exciting in that sense. Again, similar to VZV, we did see that secondary HHV6 infection reactivated quiescent HSV1 to induce beta amyloid. So again, we're kind of starting to see this trend that maybe the background of latent HSV is really what could potentially cause some of these really robust downstream effects. And this is a bacteria-derived LPS. This is a very generic molecule that's released from different types of bacterial strains. And similarly, again, we saw here that HSV can be reactivated by treatment with LPS, whereas on its own, it's not really doing very much. So this is really kind of led us to this conclusion that the initial latent HSV is really important in affecting these downstream results. And so as we all kind of know, there is a definite correlation between TBI, particularly chronic TBI or in certain populations that are more exposed to this type of repetitive concussion. They seem to be more predisposed to CTE and other types of neurodegeneration, in particular at much younger ages than the general population. So for these studies, again, we really wanted to understand if we could induce latent HSV and then exposed to some sort of injury. And again, in our lab, we have access to different types of biomechanical injury, which was quite useful. And then we wanted to, again, just kind of explore some of these rules in vitro. And what was important to us too is that this kind of concept is clinically relevant. This is just a case study of an individual who had a minor head injury and had reactivation of VZV and HSV post-injury, even though this injury was mild and essentially subcutaneous and not traumatic enough to cause any kind of skull injury. So I think this case report really gives us clinical relevance to this association between head trauma and subsequent reactivation of herpes viruses. So for these experiments, we utilized, again, this biomechanical system we have in our lab. We wanted to potentially mimic either direct impact injury using this controlled cortical impact model, which is more severe in the sense that it's literally taking these sponges and a piston and you basically punch it in the head. And this is meant to exert quite a lot of soft tissue damage, which would be comparable to having a open skull injury or a repetitive concussion. In this case, we have a 3D mimetic skull that's been printed, and the brain tissue sits within this cylinder, and you again expose it to a piston blow, but in this case, you have no direct trauma to the underlying brain tissue. And just this is not looking at HSV infection at all, but we can see that in response to these injuries, you do have very residual A beta expression, but not really much at all. And again, our goal in doing this was to really understand the role of HSV specifically with regard to TBI. So again, we took that similar model in which we, this is the same model without HSV, and this is just looking at gliosis and inflammation. And you can see here with either CCI or CHI, you see robust GFAP, which is pretty expected. In the CCI model, again, it was a bit too traumatic an injury to proceed, and our goal was really in understanding more of these low-grade chronic infections, which is why we went with the closed head injury or a concussion, as I call it. And this is also showing just that by PCR, you see IL-1 beta expression. Oops, I skipped. So just to kind of explain the context again, we take these donuts that have been seeded, in this particular experiment was for about 4 weeks. And so we treat them with either MOC or HSV for 24 hours, establish latency by treating with Valtrex, and expose to closed head injury and allow the brains to recover for about a week, and then do post-harvest analysis. And what we found was pretty striking, was that, again, this top panel here is A beta, and what I hope you can appreciate is that this latent HSV treatment followed by very mild CHI resulted in this really robust upregulation of A beta, which was super exciting. The students I was working with were really, really jazzed at this moment, which I was as well. And so we wanted to understand as well if you do a repetitive injury, is there any kind of additive effect to increasing the number of hits? And again, similar setup. We have the singular closed head injury in comparison to a triple injury here. One caveat with this is that these brains were matured a little bit longer, so it's meant to kind of mimic more of kind of an older brain. And you can see here that HSV transcripts are significantly higher in the 3-hit model versus the 1, which again was pretty exciting. And if you look at A beta here, you see a similar trend that the A beta expression is significantly enhanced in the triple model. And as well, we don't really see too much p-tau upregulation unless we have the 3-hit model. So at least in this in vitro model, we find that A beta typically shows up before p-tau, and p-tau in this case is typically a more downstream marker of post-HSV expression. And then very quickly, a reviewer had requested, and I didn't dispute it. I probably did at the time, but now I'm past it. They wanted to know what the impetus is for this sort of reaction. They wanted to understand if you neutralize this inflammatory event or prevent it, can you not only reduce the inflammation, but does this also prevent the downstream TBI-induced HSV reactivation? So for these, we performed similar experiments just in 2D in the presence or absence of IL-1 beta blocking antibody. And this is just a 2D model meant to mimic the 3D one I just mentioned. Here you can see a similar trend in which you have A beta upregulated in response to injury. And so I'm running a bit short on time, so just to kind of give the summary here. The latent HSV plus injury in the presence of anti-IL-1 beta actually reduced both gliosis and inflammation. And as well as the ultimate downstream readout in our case, which was HSV and beta upregulation, suggesting that IL-1 beta, at least in terms of TBI, is potentially what's causing this reactivation of HSV. And this is kind of our summary, which is, again, this latent HSV once exposed to injury, particularly in immunocompromised individuals or in aged brains, results in an injury that will also reactivate HSV and result in downstream neurodegeneration. And I won't go too much into this, aside from we use this model as a screening tool to understand whether or not compounds of interest could potentially be used as a therapeutic or a preventative, and these were just a few that came up. Resveratrol was one, and green tea derived derivatives. And then kind of this was our big push sort of at the end, was to understand the potential role of vaccination against VZV and the reduced risk of dementia. And for this study, again, we utilized, this is work from Randy Kaur's lab, in which he was able to clone the monoclonal antibody that's generated in response to Zostavax infection, or vaccination, sorry. Using B cells, he was able to clone this particular functionally blocking antibody. And again, similarly, just using that same setup in which you induce latent HSV infection followed by VZV in the presence or absence of this anti-VZV antibody that's relevant to vaccination. And we saw that this antibody was able to block VZV as we would expect. And what was very exciting for us, we found that similarly, it was also able to block this reactivation event that we see that we had seen in previous experiments. So I think in all, we were very excited about it, and we hope to pursue this a bit further. And this is just showing PCR, showing a similar trend. I've wasted enough of everyone's time, but a future direction, we think that possibly this monoclonal antibody that's induced from vaccination not only blocks VZV, but potentially HSV due to the homology between the viruses. So in general, I hope I've communicated to some extent some of these things we've discussed, and most importantly, from my perspective, the role of latent HSV in reactivation in Alzheimer's disease. And this is my lab and the lovely Ruth and the gaggle of people there. Great, thank you.
Thank you, Dana. That was really interesting. We're now going to move on to the panel discussion. So with Garth and Brent and Madeline and Graziella and myself. And who else? Did I miss somebody? Six chairs. Did I have five people? Fit it anywhere. Yeah,
Okay.
Okay.
So we're going to try to use the mics at everyone's seat. If your mic isn't working, just wave and we can, bring another one over. Does anyone have any questions to start off with for any of the speakers that just went or for our panel?
Don't be bashful.
Janice, can you turn your microphone on? Yeah. We're trying to talk about that. Well, they won't hear you on Zoom.
Is this on? Is this on?
Is it working?
Okay, can you hear me? Oh, that sounds okay. I'm curious as to, I don't know much about HSV-1, but what is thought to, I guess, keep it latent, or is it known what it does to react or come back out of that naturally? Oh, doesn't sound like it.
Oh, there it goes. Yeah, so I think it's typically latent most of the time. So based on what I know, it takes some sort of inflammatory event, whether that's kind of, an illness or even chronic stress. I mean, there's lots of triggers that are known to induce a reactivation event, but its kind of natural state is latent. And I think the reason it's so important in Alzheimer's specifically and has maybe gained such traction is because so many people have it and don't know it. And it's like, and it's almost always off. So you wouldn't even know you had it, but.
Is this on?
There it is.
Okay. The reduction in the immune system, just, temporarily, because I can use myself as example. I had shingles because I didn't get the shot. I didn't want to do it. So this was back years ago when the old shot was pretty terrible, and I decided I wouldn't get it. So I ended up getting shingles. I have to laugh probably over the paper with Harvey Singer. But anyway, I could feel my whole body just drain over getting stressed. And I think it's stress and reduction of, probably your innate immune system, because you're not probably reducing your antibodies, particularly at the time, but it's just enough for it to reactivate, and that's shingles. But I'm HSV1 to be in the same category. And my husband's a virologist. Do you have anything else you want to add? Michael, do you want to add anything to reactivation? I mean, the immune system is what, because our T cells too, all viral infections, cell-mediated immunity is the primary fighter when you're, closing it out and you're putting your virus away. But when you get stressed out, some of that just gets reduced. Your cytokines that are coming from your T cells. So.
Thank you.
Sorry, I'm going to read a question that came into the chat after Brent presented. So it's from Barry Greenberg, and he's wondering if elevated GFAP surrounding blood vessels in the brain in pathological sections was identified.
Yes. Yeah, we've seen it in a number of our different cases in the bank, and you'll normally see a little bit of GFAP around the blood vessel, and that's completely normal. But when you have reactive astrocytosis in that region and quite a bit of GFAP signal, that's abnormal. And that together with the perivascular lymphocytes suggests that the blood-brain barrier is breaking down in those cases, and specifically in those areas. We didn't see it in other areas.
On your talk, you were showing in pandas the perivascular cuffing, and that's also been found and reported in Sydenham, Korea. So that makes sense that all fits together. I was happy to see that because that confirms that it's at least that part is also similar to Sydenham, Korea.
And to add on that, looking at thousands of brains at this point in my career, a little bit of perivascular cuffs is seen, not in every autopsy. I would say maybe in 20% of my autopsies. Does it mean something? I don't know. Not to the degree that I've seen in the pandas cases, but I do see every once in a while a little bit of perivascular cuffing.
So we've seen that as well, Brett, in a number of different cases with that cuffing. Also, the GFAP issue shows up in a lot of the analysis of the Alzheimer brains that we've evaluated. I have a comment about the HSV1 story too, though, and a question about it, is that with reactivation, first of all, it doesn't seem like you're getting particles made of HSV1, at least not to any great extent. So it doesn't seem like the virus is actually being made and duplicated so much. But also, do you find that you have antibodies to HSV1 increasing in the blood of an individual that now is starting to have mild cognitive impairment or early Alzheimer's disease, and correlate that to the reactivation event?
Yeah, I think that's a good question. I think it goes back to the idea around IgG and does it mean you've been infected, or does it mean you've been infected and fought it successfully? So I think Dr. Duggan had a previous study in which he looked at IgG levels in the brain, and it was initially there was this thought that if you had IgG, it just meant you were infected. But now I think that the thinking is that if you have IgG, it means you fought it, and you are less susceptible to some of the downstream because your immune system was functioning properly.
Okay.
I think, I don't know.
Dana, while you have your mic on, there's also a question for you in the chat.
Okay.
So this is actually from Lavinia Alberry, and she would like to know, does the finding happen in younger brains with regard to traumatic brain injuries, and should they be taking antivirals from the day of the injury to prevent reactivation?
I mean, I think that's kind of the idea. I don't know if people here are familiar with the Columbia study. So I think so there was a clinical trial out of Columbia University that just looked at the potential role of Valtrex in Alzheimer's disease. I think the problem with that study was that they enrolled people who already had it. But I think to your point, if you take it post-injury as a preventative before there's a chance for the virus to wreak a lot of havoc, then there's a real role for it there. I think once it's already done, taking antivirals may not be as effective, I would guess.
Thank you, Dana.
Thanks.
Liz?
Okay, can you hear me? Yeah. Okay, this question's also for Dana. You said the peer reviewers asked you to manipulate IL-1 beta?
Was it you? Just kidding.
No.
No, you're just kidding. Not trying to fight anybody.
I have the same good idea in a grant, so I think it's a good one.
Yeah.
What was the rationale that they gave for IL-1 beta specifically?
Because I think that one is the most closely associated with TBI. So like in terms of any kind of head trauma, I think that's the inflammatory cascade that's most studied in head injury specifically. I don't think the other ones are, not out of the realm of possibility, but that's just the most focused.
Yeah, I guess that was my question was, did you look at TNF alpha or IL-6 or manipulating any other cytokines, or just even all cytokines, like giving a blanket anti-inflammatory and seeing if that also had the same beneficial effect?
Yeah, I mean, I know there's been clinical studies that have looked at the role of just anti-inflammatories, NSAIDs, and they have found a protective effect against dementia just as a global treatment. So I think to that point, it would make sense for sure.
I know that Sue Griffin's work at the University of Arkansas has shown that the different alleles of IL-1 beta and IL-1 receptor are associated with likelihood of getting Alzheimer's disease. So the ones that are more pro-inflammatory, you're much more have higher risk of Alzheimer's disease. So that kind of makes sense based on Sue's studies.
So should football players with herpes labialis not be playing football?
Yeah, I don't know if HSV2 is as associated with, but I think they should be careful what they do with themselves as in general. No, I do think, I think on the age-related question too, I think that's why these guys with CTE and the football players, their neurodegeneration shows up so much younger than the general population is because they've been exposed to this type of repeat injury so often. And at such a younger age, whereas, their blood, in terms of route of entry, it's not fully fleshed out as far as I can tell, but I think they could potentially have blood-brain barrier impairment earlier on than the standard population, and that's possibly why.
Recently, there has been a study that using propensity score matching that soccer players with.
This mic is kind of quiet. You have to almost hold it really close.
Okay, so I was saying that I believe that only recently there has been a study published where there's like a decent sample size where they use propensity score matching to match all the other variables that could also influence the Alzheimer's or dementia risk in football players and the regular population and the normal population, because otherwise there are the other factors that are more, physical activity or lifestyle, all those other things that are also associated with dementia risk that were not taken into account into the previous studies of football players. I have a study in the UK that's motivated by the fact that the guys who won the World Cup in 1966, most of them died with dementia. And FIFA and other organizations are funding this research where we are using this matching system with regular people So we can take into account all the other, the exposome that you mentioned earlier, with soccer players.
There's another issue there too, and Brett sort of hit upon this about football players with let's say you have cold sores, okay, and then you have a breakout periodically. If you have an immune response to that, because it's typically HSV1, okay, is that protective? Are you actually enhancing your protection by having an immune response with the breakout, so that anybody in this room, if you have cold sores and you actually develop that, it could be protective, because we typically think of it, you're associating that with also being infected with HSV1, but we're not thinking of, well, how are we really protected from it, right? So peripherally, it actually could be helpful.
It would be interesting. It would be interesting to do a study to see what kind of titers people have to HSV1 compared to all these different scenarios, so that you could maybe potentially find out, if you needed to be immune in order to protect yourself. And there would be those of us, maybe like, I don't ever have HSV1, so would I be more protected? I made it this far and haven't lost my mind yet, but.
I know that the timing of the data collection and everything may be a bit wonky, but in the study that we have in the UK, we have all sorts of health records and biosamples for these individuals. So we can actually test some of these hypotheses.
All sorts of herpes viruses you would
Oh,
look at
yes, and one
response to HSV.
Yeah. And I don't want to sound nasty to soccer players, because I love soccer and it's very entertaining. But I think that a more relevant population will be women who are victims of domestic violence, because head injury in that population is highly prevalent. So that would be, and that's something that it to be, if shown, it would be relatively.
It would be
it
would be extremely important between that and the Valtrex, because the Valtrex, I can vouch for it from my shingles, that when I got through taking it, I felt the best that I had felt in a long time, so I'm sure it took care of a lot of that, even though I don't seem to have HSV1. I may have problems with the zoster. So anyway, if I could just say, I don't want to get off on something else, but I do want to say before we end that I wanted to talk about the TH17 and the things that Mickey and I were talking about earlier, because they relate to everything, and not the dementia, because the dementia, we don't want to give people immunotherapy. And that's the other thing is people, before we get off of this, people that are treated with immunotherapy, that are given the checkpoint inhibitors and given some of the other immunotherapy reagents, are they more likely to get Alzheimer's? Do you know? It's another study that really needs to be done, because checkpoint inhibitors lead to myocarditis. And, this is, of course, another type of disease that I'm getting ready to talk about in its relation to all the chronic diseases that you all study, and what you might do. I'm not going to do everything. We might consider looking, but the fact that the strep and the Lyme and many of these are upper respiratory tract infections. I don't work with the gut microbiome, but I have a lot of experience with upper respiratory tract. So we decided to do a longitudinal study, which that is the next thing that I recommend to all of you. If you're going to find out anything in a disease, one sample isn't going to do it. You're going to have to get a study. It's hard. It's how I went for 15 or 20 years doing just human work with NIH. I dropped my animal model, and it's what cost me my grants, because then they were upset after COVID that I didn't finish my longitudinal study, and the reviewers didn't, and I'm old, so I can't complain after 35 years of money. But, It is very important, and that's why people don't do it, because they're afraid that they won't get money, because if you get in a mess with your longitudinal study. But it was so wonderful. What we did was in the first study that's published in 2016 in JCI for myocarditis, is we identified the 3 biomarkers of non-recovery. So in your chronic populations, I will bet that they're the similar ones. In other words, for Lyme, I remember when John Alcott, when we were together at a meeting, it was the Global Lyme, and they weren't working with TH17, but they immediately went back, and all of a sudden, boom, he had a paper on TH17, and there it was, just like strep. And that's why they're so powerful together, is they nearly kill children and make them so sick. And so what happened was that myocarditis is the same way. It's a Coxsackie virus. It starts out like that, but then when you get chronic disease, it's in a very small number of people. Most people get better. Just like in Korea, remember I was showing you that the antibodies go away in the pharyngitis. None of those people get sick. But the ones that get sick, what they're making is high IL-6. And they're making high IL-17A, and the IL-6 is driving all those autoantibodies. So in these type of infections, see, this is different from dementia. You would maybe hurt the people with dementia if you gave anti-IL-6, but you don't even have to give that. You can give drugs like Cellcept, even though I don't like them. People that have, like, for example, psoriasis, that's IL-17 too. I mean, all of these are connected together, and they're your chronic infections that you all are all talking about. And I think that, that type of treatment, if you're not afraid to use it, maybe even 1 dose of the anti-IL-6 or 2 doses will just take care of it. I mean, Cellcept many times for pemphigus takes care of it in just a few months, and the people get off of it and they're well. The pemphigoid, not maybe the worst kind of pemphigus, but that it's all related to these immune cytokines, and the IL-6 being the biomarker of the non-recovered people in myocarditis, or IL-6, IL-17A, and the autoantibody. So, and the autoantibody comes from the IL-6. It's so high, and it's the highest of all of them. The people that recover don't have this. They have it high for a little while, but it goes away. And so they are the ones that just don't ever stop. It's like in COVID, who were the people that died? They were the ones that couldn't stop their inflammatory response, and they ended up and die. Who are the people that die in influenza? The ones that can't stop their cytokine storm. So again, trying to use that in your chronic populations could cure them early on, and they might not have to take it for a long time. So that's it. I just wanted to say that.
Jennifer?
Turn the mic off. Is the light going on?
So my father had Lewy body dementia, and before he's got diagnosed even a year before, he had chronic eye issues in one eye and skin issues. And
Hardly hear.
okay.
Just put it close.
Okay, sorry. Okay, thank you. So we ended up saying, hey, these are all connected to we noticed that when he had his skin issues and his eye issues, his memory issues, and his cognitive
Was
issues
he a
together.
diabetic? Was he a diabetic? Is that what you said?
He was not a diabetic.
Lewy body, okay.
But he had Lewy body dementia. And so later on, we figured out that it was the Demodex mite that he had. And so we're like 100% sure that it was either the cause or the carrier to whatever caused his Lewy body dementia. Well, I was thinking on PANS or PANDA, and I recognize that a lot of the people had skin issues as well. And I was wondering, has anyone done a DNA PCR test to see if there's a vector involved? Because when we've done DNA and PCR tests on people or dogs that have had skin issues, like my dad, it was Demodex, which now it's getting more public awareness because people are actually receiving Xdemvy for eye issues with the Demodex, which last year, no one had heard of it when I mentioned, the mites. But we are certain that it is, like I said, either the culprit or the carrier. So would doing a DNA or PCR test on the people who have PANS and PANDA give you more knowledge of maybe, something that might be spreading the disease as well?
Are you thinking about a viral, just
Yeah.
finding an extra, something else, a viral?
Yeah.
I don't know. Does anybody know? I don't.
Because I know they carry bacteria and all sorts of disease.
I mean, I think here it's been very enlightening to hear about the Candida and all these different other organisms of the viral issues with the HSV1. It's very exciting. But I mean, we have not, I mean, it could be that there are co-infections. I mean, like I was saying, the Lyme and strep, I know just from years of talking to patients, that the ones that have Lyme and strep, they're so sick, they're bedridden, and no one knows what to do with them.
Mm-hmm.
And several of our cases within the brain bank do have evidence also of Lyme disease. So co-infections, I think, are very true. And we've seen a couple of talks with polymicrobial findings in the brain. And so it's not usually just one organism, I don't think.
Yes, and I know that mice can carry Lyme as well. There's a study on it. So that's the thing is if you're, if you have the mite and they keep on injecting that Lyme disease in you, then your burden is much higher than.
Well, look at the Lyme disease with the co-infections. You get Bartonella and Babesia together. I mean, this goes sometimes with the Lyme.
Does anyone else have any questions or comments?
Yeah,
Okay, I'll finish with the important question. Everyone here and, other people that aren't here, wonderful correlative data showing pathogens linked to neurodegeneration. What do we have to do going forward to kind of make this field a pillar kind of in the neurodegenerative field?
Yes. So, that's a great question. And we get asked that question a lot too. And we're all asking it ourselves is how do we prove it? How do we prove causation with these different types of infectious agents? Now, there is a trial that was just funded for Porphyromonas gingivalis with the company, Lighthouse Therapeutics, for $48 million, $48.5 million, okay, to run a trial that way to see if their drug can actually now treat for people that have that organism and would have Alzheimer's disease or prevent Alzheimer's disease, et cetera. So that's a good thing. If we try to get a clinical trial for every different organism we're talking about here, I think that'll be impossible. I think the way we're going about this, though, is trying to come up with that consensus approach of evaluating using different laboratory techniques, different laboratories doing the work, looking for, okay, what do you find? What does it mean when we find that? What are the consistencies? What are the differences? We have a lot of ways that we can evaluate. And then using models like animal models and the organoid models too, of showing that, okay, here's an infectious agent taken from brain tissue and you're putting it into a 3D model and you're showing you can generate pathology. And it's sort of like, if we have overwhelming evidence by study after study doing that, then it's just a predominance of evidence weighing forward. The other is and Nikki and I have talked about this, is actually through PR. Okay, we need to get the message out to the Alzheimer's community, to other communities, that what is being found here needs to be incorporated into the total exposome and genetic risk and included in with the analysis. So So maybe that's where I've talked to others about this, using AI approaches of curating the data so that we're putting this information in. There are thousands of reports of what we've been doing with infection and Alzheimer's disease. If that's included with other analyses, I think what will fall out is that this is the missing link that has never been given the credence that it is warranted.
So I think actually that we're kind of at a tipping point in terms of PR. I know, like when I first started in the field about 10 years ago, I would just get laughed at. I was just invited to the Alzheimer's research meeting in Berlin, Germany this week. And we're just accepted as part of the mainstream, at least over there. But most of the speakers were actually from the States. So I didn't get any pushback at all. It's a completely different environment now than it was a few years ago.
And I like to add to what Brian said. And I think that the triangulation of evidence, that is a very common approach, and it's even like a philosophical approach to science. The triangulation of evidence, where we do like clinical trials, we do lab work with mice, humans, and then we look at also like human studies, like longitudinal studies, where we can take into account all the factors that are also associated with dementia risk and so on. So that look at, it's almost like a kaleidoscope, that toy that our kids play. For me, it's almost the same, right? We had to look at this situation, at these associations, right, from different perspectives. And if we find robustness, it would be fantastic. And if we don't find robustness of evidence, then we can also generate new hypotheses. And also from the population perspective, I don't think we should even expect robustness because of the diversity and the genetics and the exposome and all those other things, but we have to understand what's happening within the context of the different populations. So my take is that approach is also contributing quite a bit.
I agree. Many of us talk about this like, okay, you have different organisms that can cause pneumonia, right? It's well accepted that that can happen. And these are respiratory organisms and many different ones. You can have parasites, you can have fungi, bacteria, viruses. Well, why do we think that that can't happen in our brains as well, and having different organisms? And that may be why we have different presentations also with all what we consider Alzheimer's disease, which I think is more of an umbrella because you have different representations there as well in that diagnosis and in the other diagnoses as well. And this goes to the life pattern here of early life to late life. And we have, I've been also calling things like infectious imprinting of early life, puberty, late teen years, late 20s, mid-age, late age. And if you think about it, look at the different types of diseases that show up, which Nikki showed on her slide, of that aging population from early to late, right? Autism early on, obsessive compulsive disorder, schizophrenia, multiple sclerosis, ALS, Alzheimer's, Parkinson's. And in some ways, it sort of makes sense that there would be these critical periods where if you're exposed, you're now at great risk for, unfortunately, for disease.
Our perspective, the type of work that I do, that is called life course epidemiology. Right? So there are different models, but the key to getting those answers is also having access to data that allows us to test those hypotheses. With electronic health records, for instance, that's an opportunity, right? But it's incredibly difficult to access electronic health records. Mm-hmm. Large data sets like the UK Biobank that some of the presenters talk about today. It's an incredibly selected. Yes, it's a fantastic data set, 500,000 people, but incredibly selected. White middle-class British people. Right? That doesn't translate to other populations. But still, we are getting there, I hope.
I wanted to interject a comment from the chat. with regard from Terry Sainer, thank you, Terry, the use of lithium orotate, which has antimicrobial properties as well as other properties. I myself have it in my supplement box already because for many years from naturopathic doctor. What does everyone think about that with regard to Alzheimer's disease specifically? Is everyone familiar with the new data that came out? Oh, it's an animal model suggesting that lithium orotate could be effective in the treatment of or prevention of Alzheimer's disease. And it is a supplement. I know some organizations have called for trials. I know the Alzheimer's Disease Data Initiative identified potentially, testing lithium orotate as one of their bullet points recently on a social media post. So that's cool and interesting. Does anyone in the audience know more about it? Mickey, any of the other researchers that are out there? Herb? Lithium orotate? Yeah. Do you know more about its mechanism? I have to say I don't. I haven't read up too much on it. I just trust my naturopath and take it. Can you, oh, well, Herb said he thinks it's anti-biofilm for everyone on the chat.
And I
You have
was
a question?
I have two questions / comments. One is hormones. How do they affect how people present with these different things or obtain these different things that we've discussed? Lyme, pandas, dementia. Lyme, pandas, dementia. How are hormones affecting those things? I think they definitely do.
I would just really love to chime in on this one because we just launched a new initiative at MIT on menstruation science, and the goal is really to look at these different sex hormones to try to understand why, at least in the IACIs, with long COVID and with chronic Lyme, you have a 70-30 female to male skew. And it's been shown that within that, the women with these illnesses have lower testosterone levels than women without these illnesses. And, that definitely, I think, raises this enormous question in the context of infection, but I also think about this in the context of vaccination and other stimuli for our immune response. Is we know every single immune cell has androgen receptors, estrogen receptors, progesterone receptor, and we have no idea what they do. We have no idea how they're controlling the immune system. And so we don't even know to tell women, does it matter at what stage of your menstrual cycle you were infected or vaccinated in terms of, the outcome or adverse events. And so I think it's very, very clear that these sex hormones have a huge impact and we have to incorporate it into study.
So this was a topic which was brought up repeatedly at the Berlin meeting that I was at, and I hope I get this right. There were at least 3 talks, and what they were doing is they have narrowed it down to follicle stimulating hormone, FSH. And as women become perimenopausal and the ovaries start shutting down, FSH levels go up to try to compensate. And so what they did, 2 different groups took the mouse models, the pig mouse, and they also took them up, and they did is they bred them so they knocked out one of the FSH alleles, and that reverted those female mice to then having the same rate of development of Alzheimer's like as the males. And there were 3 different groups that were doing these studies, slightly different variants. They were all coming to the same conclusion. So I would just encourage people, I just learned about this like a couple of days ago. Look at the literature on FSH in Alzheimer's disease.
I've been fortunate to receive some funding from the Alzheimer's Association to look into hormones and brain health. And we are also hoping to get additional funding with the team to examine this in the context also of infections. But again, I love drawing these diagrams that allow us to understand the different links between the different variables and players in the picture. And it's a very complex topic because we have, genetics playing a role there with the infections, obviously with brain health. We have the hormones, whether, for example, menopausal women take HRT or don't take it, the timing of the intake of the adoption of HRT. So all those factors are in the picture. I can email you the diagram that I have looking at the different links. And again, the amount of data that we need to really look at the full picture is, at the moment, my impression is that at least at the population level, we can put different pieces of the puzzle together, but we are still a bit far from looking at the whole interaction of things.
The other thing that they noticed with these mouse models of with the FSH, when they knocked out one of the copies of the genes, that then the mice, the female mice, also did not suffer from osteoporosis, nor did they develop obesity. In fact, their fat converted more from white fat to beige fat. And so there was this kind of consensus, amongst the people there that these elevated FSH levels, are really, driving a lot of the increased pathology that you see in postmenopausal women.
So would women postmenopausal be advised, and would it be advisable, and I know this is a new area, to continue to take hormones so that they don't have these issues?
So one of the studies that's going on is one of the groups is actually developing a monoclonal against FSH. And so they want to actually, their goal is to develop this actually as a therapy, because FSH will cross the blood-brain barrier. So if you can just reduce the levels in the blood, it will reduce the levels in the brain. And the other things they showed is that all throughout the brain, there are receptors for FSH.
I think we should probably honor our poster presenters that are getting ready for us upstairs also to have some of those wonderful antioxidants in the red wine that we saw up on the screen, which I thought was very timely. I think, Lynn, was that you or Dana? Dana, did you put red wine up there? Okay, there's also red wine upstairs, incidentally. Thank you. I apologize to everyone online who can't have red wine with us. I just want to close with a few remarks. Thank you to all the presenters, all of the attendees, all the families, all the loved ones, all the patients, and everyone that's not here. We thank all of you. And up on the screen, one more thing. So earlier, Eve mentioned that we have a special issue with our AlzPI colleagues at Frontiers. We have a second one as well, thanks to Editor-in-Chief Dr. Barry Greenberg, in Translational Research and Clinical Interventions, which is an Alzheimer's Association journal entitled, which is very perfect for Kevin's question, Infectious Agents, Causal or Risk. So 2 calls for papers. The QR code takes you directly there. Thank you so much, everyone. Thank you to the Osteopathic Heritage Foundation. Thank you to the tech team today, to OU, HCOM, the wonderful panel of people here today, and everyone that presented. I count you as my superstars, as many people have said today. Thank you.